Yeah. We can also move this panel so that we can see Marco. Do you want this?
Yeah, this is all good. Thank you.
Is it okay so that we can see Marco?
But I can see you.
Okay, great.
Oh.
But I will have to increase my volume because she will speak right here. I hope it's here.
All right. Good morning, everybody. Good morning, everybody. Thank you all for coming up here on Saturday morning after the conference, the workshop. I'm so happy to see everybody here. I want to take this opportunity to reflect on where we started and what's our objective of putting together this workshop on next generation adaptable computing for omics. We started last year, Pepe and I put this together with the objective of bringing in the community from the bioinformatics, the computational biology, and all the folks in the life sciences that are involved in amazing work that is life critical. They're working constantly on algorithms that impact lives in it could be in the neonatal ICU, it could be in the neurology, neuropathology, in neurosurgery, etc. I mean, it's amazing the kind of work that the computational biologists are at, and we folks at the FPGA community constantly talk about workloads and applications that are latency sensitive. We talk about energy efficiency, and we want to, look at workloads that are diverse. And this is an opportunity to look at the workloads that are of interest to the computational biologists. They talk about the genomics pipeline, and they talk about, you guys talk about latency sensitivity and how bringing down the sequence analysis from hours to minutes could make an impact on life-saving opportunities to patients. And if you could provide diagnosis to the cancer patient who's waiting for what could be the opportunity that is available in a few hours than in waiting for a few days, I think that's, for us FPGA designers, it is fantastic to make an impact on saving lives. Having said that, we talk about FPGAs being very versatile. We want FPGAs to be used across application domains, and we are very interested in applications that have low latency and latency critical. So why not look at this? So we put together this workshop last year and deliberated on what could be the next steps. And this year, I'm so thrilled to have Onur, Konstantina, and Marco, who's joining us online. He's online this year, and he has promised to be in person next year. And we want to do more and take this forward. And I'm very happy to welcome folks from that have come in here. Yeah, so that's the larger objective of bringing together the community. What could we do to do more with this community? Is it about looking at problems that make them available? Is it benchmarks that we are missing? What is it that's stopping us? It looks like a very unique combination of what FPGAs can offer and what is needed in the bioinformatics space. What is it that's stopping us? I would like all of us to take a few minutes to think about this and how do we take it forward from here? We are building a small community here. How can we nurture this community to move forward and make an impact in these lines? So as Marco has also expressed interest in taking it forward and maybe have a workshop series, have a call for papers moving forward. With this as the at the outset, I would like to request Marco to take over. Marco, can you hear me?
Yep, and I'd love to. but I just send a request because I cannot yet share the screen.
I can hear you. Go ahead, please. Or I can move the slides if.
There we go.
Yeah, good.
So I hope you can see the screen.
Just a moment. Maybe you can try again, Sari.
Oh, it's shared.
Okay, good. Okay.
I was on the same slide that Madhura was.
Okay, yeah,
Okay.
go ahead.
So let me see. No, unfortunately, nothing is happening there. So I don't know if you are able to see my screen. Because I can see the slides projected.
Yeah, maybe then try sharing once more and we'll get there, I guess.
Okay.
You allow you there?
Yeah.
Okay. And I just need to change the screen.
I can see a rhino.
Yes.
Now I can see part of
Yeah.
the share slide.
No, wrong slide, sorry. I'll get there.
There we go. Let me see. Yes. Now it's me.
It's great. Where is the full screen?
Left, top left, green button.
Which one?
Left.
But I think David is speaking, but I think that they are already the ones that was sharing. Okay, great. Well, I'm sorry I wasn't able to be there, but as Madhura said repeatedly, I'm going to be there next year. So thank you all for being there. It's kind of weird to talk to the podium and not from the podium, but I hope you are going to enjoy these 5 minutes more or less journey into what I was willing to share with you. So first of all, as Madhura did already, but I do really want to thank Madhura, Konstantina, and Onur for all the work they did to put together this workshop. And I'm just thrilled to have the chance also to move it forward also for the next year. So because of this, I was just kind of trying to, I was interested to play a game with you. Now, unfortunately, I don't know how many of you are there and how you are dressed, but I hope you're going to be able to follow me in this journey. Oh, thanks. Now I can see you. That's great. Thanks. Um, Next generation of adaptable computing for omics. So we are all in the FPGA. So I was not really willing to describe the rationale behind the reason why we want to go and use FPGA, obviously, because otherwise we had a problem. But the idea here is to kind of try to focus on what adaptable means. With the challenges that we have to face when it comes to work with adaptable computing infrastructure. So they have to be goal-oriented, they have to be approximate, and they have to be fast. Now, the kind of interesting things with respect to the domain that we are targeting in this workshop, omics, is that all these three characteristics are definitely of interest, of great interest, and not only for adaptable computing system, but also for omics. So let's try to see why fast. Well, as I was mentioning, I was kind of willing to have a kind of an interactive, since I'm not there, at least I was kind of trying to make it as if. So I don't know how many of you are wearing black t-shirt, but I know that at least there were a couple of wearing white ones. What about jackets? So I think that there are a few among you who are wearing jackets and maybe others that are not. Maybe skirts, why not? Ties, maybe not the case. I cannot spot anyone wearing a tie, and maybe heels. Just a very super simple example. Sneakers, we can go on forever. So we can have the entire workshop just listing these things. But the idea here is that we are all different. And the kind of funny part is that we are different in the way in which we are dressing. But what about the way in which we are treated when it comes to care? And when we are not feeling good, the way in which we are treated most of the time is basically the same. There are a few tweaks here and there, maybe because of the weight that we have, maybe for the age, but not really. We're not yet to the point in which we are really creating something that, as we all aware of, is called personalized medicine. But these things are definitely useful. What is somehow preventing us to be able to do that is being able to provide necessary information on time. So it's not really something that cannot be done. And there are tons of different examples. Here I was just taking the one from cancer, in lung cancer, because we do have some expertise in that domain. And we know that being able to provide not maybe just the cure, but being able to spot it in advance can make it really the difference. And being able to do all these things is because of the ability of being able to process an insane amount of data fast. And this is quite a key point in the research that we are doing. We come from the FPGA's domain. We know that we can process data fast, we can be efficient. We just have to target the proper domain and the proper way of doing things. We may need to be approximate somehow because being faster has some kind of constraints, and we do have to be able to provide fast results that may not be as accurate as we may be willing of having. And that is also interesting because when it comes to omics, we still want to have medical doctor in the loop. So we may not be willing to have the final decision. We want to have and keep the human in the loop. And that is also going to allow us to kind of try to find a way in which we can tolerate some sort of approximation. And that's also very important when it comes to kind of a situation in which we are going to take the human as the central focus of the research that we are doing. We may have also questions like this that have to be answered. How do we feel? And it's not just a matter of our feeling per se, or we are happy, we are sad, but this may also affect the way in which we are going to respond to certain treatment. So this is great. And this is also another chance because this is another thing that is going to be really super tough to be computed, to be processed properly. And this seems to be quite sort of unrelated to the things that we are doing. So when we are thinking about omics, we can think obviously of genomics. We can think of data that are out there belonging to other source of data, like also imaging, and combine them together. But also feelings is something that we may want to have an eye on, because the way in which we are going to respond, as we were previously mentioning on treatment, may belong also to our emotional state. And this is something that is going to be quite on the edge of few research that are coming out nowadays. And also in this domain, we can have a lot of interest with respect to the community that we are from, using FPGAs. And well, we know that sometimes using FPGAs can be hard and can really involve our feelings. But in this case, it's a kind of a different context, obviously. But there are a lot of things that can be useful in this context. We may also try to see how to use machine learning technique within a sort of an explainable AI context, because in this case, we want to know and we want to be able to provide information on how we were able to achieve a certain result or a certain answer with respect to the context in which we are. Because as I was starting from the beginning, we still want to have the medical doctor in the loop. So we need to provide them tools that are going to provide answer fast. Those answers may be approximate. And because of this, we need to provide them also a kind of a key of thoughts in order to be able to allow them to understand, to reconstruct the reason why we were suggesting them to go in a certain way. And at the end, we have to be goal-oriented. Being goal-oriented means also to be specific. And with respect to the overall context that we are all, well, sorry if I'm saying we are here for, you are here, I'm here online, but we are here as a community. We have to be goal-oriented, and our goal is to be oriented on omics. And I think that there is nothing better than the workshop that Madhura started. And I'm really thrilled to have this set of talks today, in having the keynote from Onur, in having accelerating bioinformatics from Yatish, and then after the coffee break, the remaining 5 talks that are going then to to provide the core, the body of the workshop. So sorry once more for having missed the chance to be with you physically in the location, but I'm there in spirit, and also because of the work done behind the scene in organizing the workshop. I'm just thrilled to provide the opening of this workshop. So thank you all once more. And Madhura, if you want to continue from here and introducing Onur for the keynote.
All right, thanks, Marco. All right, I call upon Onur. Onur needs no introduction. I'm very thrilled that you're here today, despite your busy schedule. Over to you for you.
Thank you. Good. Windows. As while Konstantina is setting up, I can say a few things. It's great to be here. This is my first FCCM. I never attended FCCM before, although I read a lot of papers and I know a lot of people in the community, I think. It's good to be here. I was invited to give a talk last year by Pepe and Madhura when they organized it in the previous FCCM. The location and timing didn't work out last time. I think this time it did work out, so glad to be here. And glad to be part of this workshop. I think it's a critically important topic. I'll give you my perspective on a small part of the omics domain, which is genomics, that we've been working on for some time now. And hopefully that'll give you some idea. I think there's a lot more opportunity in this area for sure to do very interesting work. It will be very impactful, I think, going into the future also. Even though it may not be the application of choice by manufacturers or system designers today, I think people will realize more and more the importance of omics and health-related applications going into the future. And there will be clearly a lot of machine learning incorporated in it also. So if that's what will motivate people, we'll take it, you know. But I think there are broader aspects of omics and applications related to life and health sciences that are going to be a lot more important in people's lives. Okay, I'll start with that. How much time do we have? Because I have lots of slides. I can talk a lot about this topic. Okay, sure. Until 10? 10. Okay, yeah. And do we have all the speakers here? Okay, there's a reason for that question, but hopefully, yeah. Okay, that's good. Sure. But feel free to ask questions also. I'm going to give you a perspective of the journey that we've been taking in this area. I essentially called accelerating genome analysis via algorithm, architecture, co-design. And essentially, as I said, system design for bioinformatics is an important problem. It's a critical problem. It has very large implications on science, medicine, society, public health, and also personal implications related to probably many of you who have been, I'm sure you have seen a lot of health-related things. And if not, COVID is probably the best example of this, actually. I'm going to mention that also. And this talk is about a very specific aspect of bioinformatics. Bioinformatics is general. Genome analysis is a small portion of it. And even within genome analysis, we're going to look at a very small aspect, which is read mapping. And I'll also talk about some of the other parts of the genomics pipeline, like base calling. But maybe some other speakers will talk about base calling and how to eliminate it, like John, who's sitting in the back, who was my PhD student. He's an assistant professor at the University of Maryland right now. Okay, so there are many bottlenecks that exist because we have huge data in this area, and accessing and manipulating huge amounts of genomic data causes a lot of strain on our computing systems. And general purpose computing systems is not the way to go in general for these, although people have been dealing with that. That's why I think this workshop at FCCM is a very good example, because we're going to talk a lot about FPGAs in my talk also. And I'm going to talk about many recent ideas to accelerate read mapping. And this is going to be more of a personal journey since September 2006. And I'll tell you how it kind of started. Like one of my friends who was at University of Washington Genome Sciences Department at the time in 2006. I was at Microsoft Research at the time, forming the computer architecture group. He approached me and he said, they're building this read mapper to be very comprehensive. I'm going to talk about read mapping, etc. And essentially, they wanted to accelerate it as much as possible. And I'm clearly very interested in acceleration methods, software, computer architecture. So I helped them by using a lot of simple techniques like compression, etc. And we achieved like an acceleration of 10x, maybe more, in average use cases. So that was very good. And I was always interested in life sciences in general. And we were brainstorming a lot of things. And we said, this is an area that will become bigger into the future. Can we actually design embedded devices, for example, that people can use such that they can do these things privately, or doctors can use at least, maybe not general people. And you can get these analyses within real time for critical conditions that Madhura earlier mentioned. There are many questions over here that you need to answer, like which of these DNAs does this DNA segment match with, assuming you've sequenced things first? What is the likely genetic disposition of this patient to a drug? And this could be done in a very time-critical condition. What disease or condition might this particular piece of DNA or RNA associated with? And how would it potentially evolve, like if you want to do outbreak prediction, for example? And there are many, many other questions over here, right? And clearly, there's even more than that. You can actually understand species evolution. You can predict presence or relative abundance of different things in a sample. You can rapidly surveil disease outbreaks. And you can develop other personalized medicine approaches and many, many other things that you cannot see over here. It's kind of cut off, so let me see. Okay. What is this? It's a oh no, it came back. So did I just close it? Okay. That worked. It didn't close the entire machine. So we wrote a lot of articles about this. I'm not going to go through these articles in detail, but this is an area that's not easy, I would say. Like when I first started, I read these bioinformatics papers and I understood nothing. Because these papers are not written for people's understanding, from my perspective. People in bioinformatics domain write these papers with a lot of jargon. There's a whole lot of jargon that you need to sift through. I still don't understand a lot of these things. That's why we wanted to write things that are more understandable to humans, let's say. And we wrote these articles. Humans being computer engineers, computer architects, let's say. Bioinformatics folks may understand. Yes? Oh, I'm sorry. I cut off the internet. That's crazy. Okay. So that was the wrong thing to do, huh? Okay, I can say things that are not recorded right now. Yeah. So when I say actually these papers are not written to be understood, I'm not joking. You know, I tell I give these papers as examples to my students on how to now not to write papers. That's why we wrote. Okay, you're back on the internet. Okay. Okay, yeah. That's why we wrote some of these papers to help other people get into the field. And I hope it'll help some of you if you're interested in this area. Okay, all good. Thank you. I'm not going to touch things that are not related to my presentation. Having said that, I won't touch this one. It's not shared still, okay.
On Zoom, it's there.
On Zoom? Oh, not on the left. Oh, okay, so it'll come back. Okay,
Good now.
all right, thanks, yeah. That's okay. Okay, so these are some of the papers that we wrote in this area. You can see that FPGA-based near memory acceleration is an important part of it. And this is one of the more recent overview readings that we have. Maybe a new one is coming with John in the future on something important. But this is a very short summary that we have at DAC. This is probably the most recent version. So the problem is we have very special purpose machines that generate genomic data. This is one example of those machines. I'm going to give you many other examples. Some of these are huge, some of these are small, but they're extremely specialized in what they do. And what we're doing is we're moving a lot of data out of those machines, and we're using general purpose machines for analysis. There are two problems with this. This is clearly very fast because it's very specialized, and this is very slow because it's not specialized to what it's processing, right? We want to solve that problem. And the second is large amounts of data movement. Basically, we don't want to move data. If you want to process data, we don't want to move data. I'm going to get to that toward the end of the talk. And this, if you actually do this, this limits the application scope and accuracy because If you want to use general purpose machines, it basically takes a long time. And if you're doing some analysis, some urgent analysis, for example, that's really necessary, that needs to be done within minutes, for example, in some cases, you cannot do that. And in the end, my takeaway message is that we need to co-design across the stack, algorithm, architecture, and devices. So let me jump into the agenda. So we all have these things. Many species have these things. And in the end, what we do is, in sequencing, we turn them into some sort of text. Later, we're going to deconstruct it. We're going to try to eliminate that turning it into a text step and try to process the signals directly. So John will have a talk on that topic on lost in translation, for example, right? So the goal in DNA sequencing is find the complete sequence of this text, ACGTs, in an organism's DNA. The challenge is there is no machine that humans have constructed so far that takes the long DNA as input, and human DNA is 3.2 billion base pairs, and gives the complete sequence as output. So you cannot get the complete sequence. All sequencing machines are choppers. They chop the DNA into some pieces randomly or not so randomly, and you need to put them together first. And that's the read mapping problem, essentially. So these are some of the choppers that people have designed. This is not a very up-to-date slide, so there are newer ones also. You can see that they come in all shapes and sizes, and they all produce data with different properties. So these are some of the high-throughput sequencers. So we're going to talk a lot about this particular one because this is very interesting. When we were talking about like embedded device in 2006, 2007, this thing was not popular. Oxford Nanopore Technologies designed this nanopore sequencing device, which is essentially a portable device to do the sequencing in the field, let's say. So this makes it easier to actually do embedded analyses and portable analyses. And we do have works in this area. I'm going to talk about these. So if you look at these devices, they have a lot of differences in terms of what kind of reads do they produce. Reads are basically how do you chop the genome into smaller pieces. A read is a string of characters, essentially. Some of them are short, maybe 150 base pairs. Some of them produce long reads. The device that you saw, Oxford Nanopore, for example, it could be 50,000, it could be 2 million. And these are also variable lengths. The issue is there's a trade-off in general between the length of the read and the accuracy that you get out of the machine. Accuracy meaning how accurate is the identification of this sequencing device in terms of identifying the base pair. It may think it's an A, but it may be actually a C, right? So with shorter reads, especially with Illumina type of technologies, image-based technologies, you can actually get a much higher accuracy. The accuracy of the longer reads have been improving, but at a higher cost potentially. So there's still some accuracy gap. So today we're in a much better place though, in the past. So genome sequencing pictorial looks like this. You have a large DNA molecule, you chop it into pieces of some different length and accuracy, and you get these reads that are those pieces, small DNA fragments essentially. So the problem I mentioned earlier is we need to reconstruct the entire genome, or at least the parts of it that we're interested in, from many sequenced reads. But even knowing the parts of it that you're interested in requires some understanding of how these things fit together. So read mapping, more formally, you want to map many short DNA fragments or reads to a known reference genome with some differences allowed. Essentially, you're mapping it to a known reference genome. That known reference genome is constructed already with some other techniques. This is called assembly, and we may get into it also. That's also important. But let's not step ahead. So DNA logically looks like this. It's physically like this. A machine chops it into these reads, and you want to map where those reads come from a reference genome. So a reference genome is really a reference. It's not exactly what you have chopped into pieces. You're just using the reference genome as some reference so that you can reconstruct what may be going on. If your reference genome is completely different from this species that you have sampled from, then you're not going to get a very good composition. So usually humans have reference genomes, for example. There used to be a fixed reference genome. I think that should be broader, because even a fixed reference genome, none of our genomes match each other. There's some very high similarity, 99.9, for example, but the rest are what may be important, because those are the parts that actually may be really interesting to examine. Okay, so you map it to a reference genome. So there, by nature, there will be some differences from this reference genome, but you want to really map it as much as possible closely so that you get a good understanding of what you have sequenced. Okay. So this is challenging because you're really mapping billions of small base pair reads. And because of other issues like inaccuracy in the machine, you do this multiple times. You need to actually, you don't do it once, you do it multiple times. And based on those multiple times, you build a consensus and you decide which one actually matches. So it's a little bit more sophisticated than what I just showed you. So there are other issues that are also interesting. We will talk about metagenomics later a little bit. Sometimes you basically want to sample the environment, right? Like I take a sample over here to figure out what's lurking in Georgia Tech conference center, which I don't want to know. There may be cockroaches, etc. Who knows? You sample this and you want to understand what kind of species are there, right? You don't know what it is, so you take it and you map it to essentially a database. So there's not a single reference genome, but there may be millions of reference genomes. And you don't know exactly what these reads belong to. The idea is to classify these reads based on comparison to like hundreds of terabytes of databases. So this is called metagenomics, and it's a much tougher problem, I think, than a single genomics. So when we talk about these, we actually need to go into much bigger storage spaces. So we do a lot of work on storage and in-storage computing to accelerate metagenomic analyses. So the entire system becomes interesting, I think. Okay, so let's take a look at some examples of this. These are some of the terminology that bioinformatics folks like. They like naming their files FASTA, FASTQ, slow, etc. I don't want to talk about terminology here, but basically there's a reference genome stored in text, and there's a sequence reads stored in some other file. The interesting thing is they don't talk about it. Once they name the file, they assume everybody knows the file, FASTA file. You got to remember it, you know. That's the part I don't like about the papers that I read. Okay, basically you want to find the sequence reads in this file, right? And for example, this blue part matches two potential blue parts over here. This green part matches these green parts. This red part matches these red parts. The problem is it's not exact matches, right? And what you want to do is really sequence to match together, right? Likely this blue, green, and red matches this part, but you don't want to actually compare everything because it's a very expensive comparison, as I will show you. You want to actually sample the comparisons and index. You form an index of the reference genome and find which parts of the sequence reads matches and which one forms a consecutive region, let's say. Okay, this will become a little bit more clear. If you want to do base-by-base comparison, this is what you would do. You compare the read that you get from the sequence machine to a reference genome. You compare every single base and you get some number, and you say these match, right? So what we do is we really do, this is an edit distance comparison because we want approximate comparisons. And edit distance, which has been defined a very long time ago, 60 plus years, is defined as the minimum number of edits needed to make the read exactly match with the reference segment. We're doing string matching essentially. And edits can be insertions, deletions, or substitutions. I like showing this with like real text. Like if you want to, if your reference is, let's say, Switzerland, and if your read is Netherlands, if you want to match Netherlands, if you want to make Netherlands match Switzerland, you actually need to have a deletion over here in Switzerland, and you need to have mismatches at S, W, and Z, and you need to have insertion. So this edit distance computation is expensive because you need to actually do this computation. It's not a simple string comparison. It's a comparison to figure out the minimum number of edits that you have. Okay, and all of these may have different meanings potentially from a medical perspective, for example, whether you have an insertion or deletion, etc. Okay, so let's look at the computational challenges. So you want to find many mappings of each read because a short read that you chop may map to many parts of the reference genome with some differences allowed, right? The question is, how can we find all mappings efficiently? You want to tolerate small variances and errors, and there are two types of errors. One is coming from the machine, and which is the sequence errors over here, and the other is subject's DNA may slightly differ from the reference. In some cases, may not slightly differ, may differ a lot. So how can we efficiently map each read with up to E errors present? As E increases, this problem becomes actually much more complicated and complex. And in some cases, in some use cases, you want E to be high because you want to allow larger differences. And this is an area that I think even the bioinformatics folks don't quite understand exactly what that should be. This is kind of put as an input to the read mappers. And you want to do this very fast. Basically, you have millions to billions of reads. And the question is, how can you design a much higher performance read mapper? Depending on the analyses you want, this may take a long time. Okay, I will not go through this in detail, but people have used dynamic programming to really do this edit distance computation traditionally. Probably you've seen dynamic programming in the computer science courses. And there's a lot of data dependencies in dynamic programming, and you normally need to compute entire matrix, even though strings may be dissimilar. Although there's recent work in this area that tries to reduce the computation that you have, which we may get to. And people have actually written a lot of both practical and theoretical papers on edit distance computation. This is one of the nice ones that describes basically why this is a very challenging problem theoretically. If you're interested in theory, you can take a look at it. So if you do this the naive way, you want to do pairwise approximate string matching. You want to do edit distance comparison. You take every single read that you have and you compare to every possible location in the entire genome. Clearly, this is not good because it's just too slow. And maybe earlier versions of read mappers did it, but they don't do it like this anymore. So existing read mappers have a lot of software optimizations, most of which I will not go through, but some other talks may go through. So existing software is designed to quickly and accurately reduce the search space as much as possible. What you do is you don't take the entire read and compare it to everywhere. You index the reference genome, and you want to actually, so you basically do a processing step that's offline. You index the reference genome, and this index is formed based on some what's called k-mers, essentially. You have strings of length k, and you use those strings of length k, which is part of your read, to index and figure out which locations contain that small substring of length k. And after that, you do some more things that I will not again go through. There's a lot of terminology over here, but you can read some of the papers that I mentioned. Basically, the idea is to eliminate as much as possible the comparisons that you do, right? By doing indexing, by doing simple mechanisms to actually filtering out what doesn't match, for example. And eventually, you want to do this dynamic programming-based edit distance computation to things that you're really not sure about. So there's a lot of software filtering that goes through. We're going to do hardware filtering and hardware support for this soon. Hopefully, that's clear. You want to perform costly computations only for a few regions that you think there might be a match. You want to quickly eliminate everything through software that doesn't match. So that's the read mapping, basically. And there have been a lot of techniques that are developed for all of these different parts of read mapping. This is a paper that we wrote that covers essentially more than 100 read mappers and different ways of doing different things. If you're interested, this doesn't cover things since 2021, but it's still a good paper to go through to understand what has been developed. It's fascinating because people keep developing new things. This doesn't cover some of the really interesting techniques that people have developed, like the wavefront algorithm, because that has been developed after 2021. Okay, so we're going to talk about accelerating read mapping. You can clearly accelerate all of these parts. You can accelerate indexing. You can accelerate filtering mechanisms. I'm going to talk a lot about filtering. And you can also accelerate alignment. In fact, there's a lot of work, old work also, that used FPGAs to accelerate alignment mechanisms, dynamic programming, Smith-Waterman algorithm. So that's been around for a really long time. And I would encourage you to take a look at those works. We cover some of those in these papers. Okay, so essentially, we want to accelerate read mapping so that we can accelerate other things that come later. Because read mapping is an initial step. After this, you do a lot of other things to really understand what's going on, like call the variants, figure out which parts actually differ, and then maybe you try to discover something related to the patient or something in science. Basically, we're bottlenecked by read mapping. There's a lot of work that shows this data. It's becoming better with some of these filtering approaches. So you don't want this sort of bottleneck in the analysis system in general. So it boils down to what I just said earlier. So we do have courses in this area. I will also use this to pitch something Yatish is organizing, and we're organizing at Recomp next week, actually. This is in Thessaloniki. This is kind of like a sister workshop on a different community. The Recomp is the major computational biology conference, and we organized the first Recomp ARC with John in 2023 when it was in Istanbul. Now we're going to Greece, the other side of the Aegean, to organize Recomp ARC. So if you're interested in this area, and if you want to interact with the bioinformatics people, this is a good venue to submit your future papers to. It's kind of like a sister thing for FCCM, I would say. Okay, now let's talk about acceleration. So acceleration is interesting because you can clearly do a lot of things in software, and people have done I'm going to talk briefly about that, but I'm going to focus a lot on hardware because that's something really important. So algorithmic acceleration is, you can exploit different things. You can exploit structure of the genome, you can exploit existing instructions in the system, and we're going to do some of that. And for hardware acceleration, I think we have to move to specialized architectures because this is, you need to match the throughput of a specialized device. And the only way you're going to match this throughput of a specialized device is by specializing the architecture to what it produces. And I will, if I have time, I will talk about processing in memory and storage. This is actually, because of the data moment, I think there's a lot to do over here. And I will try to conclude with some future opportunities like new technologies and applications, because applications are also evolving, and also technologies, sequencing technologies are evolving, and I think we need to adapt going into the future. So this was our first step. This was the paper that I kind of mentioned earlier that we wrote first after optimizing the mapper that they developed at University of Washington Genome Slicing Department. It was a very comprehensive mapper. The goal was to tolerate up to E errors. Nobody used it these days because it's very hard to also change the space. One of the issues that you will encounter in bioinformatics is it's really driven by people who use software and who don't want to change anything in software. So it's good to think about that. How do you actually get things adopted is going to be important into the future. Maybe I'm joking by nobody used it. Somebody used it maybe, but I don't know. It's not generally used today. So we actually did a lot of studies to understand where time is spent in this mapper and some other mappers that are optimized for in various ways. And we found out in one of these studies that more than 60% of the read mappers execution time is spent on sequence alignment. So we want to actually accelerate it. And our overarching key idea on the next several works that I'm going to talk about is to do what actually people have been doing, but also in a very hardware-friendly way, such that we can accelerate in FPGAs and other things like SIMD instructions. And the idea is to filter fast before you do edit distance computation, because we want to minimize these very costly approximate string comparisons. So if you want to look at a pictorial, you have some sequenced reads. Some of them are too dissimilar to the reference. Some of them are similar to the reference. You want to quickly figure out which ones are too dissimilar to the reference so that you don't look at that. You want to focus your processing power on these things that you think are similar to the reference. And ideally, you would like this thing to be perfect, but if you want to make it perfect, then you get closer to the computational complexity of this. So we want to do very simple computations to identify which ones are dissimilar to the reference. That's the key idea. So we actually started with software. We figured out that the software was not good enough, so we did software filters that I'm not going to talk about that exploit the structure of the human genome, for example. You can take a look at that. I'm going to talk about hardware-friendly filters that we developed after that. This is one of the works that we published at Bioinformatics. And the idea is, if you want to do string comparison, the simplest thing that you can do is really Hamming distance computation. I'm sure some theoretician will figure out that that's what I said is wrong. But at least from my perspective, you compare every character with every other character. And for example, if you have Istanbul, one of my favorite cities I went to high school in, you want to compare to, this is, let's say, your reference, and this is your read. And if your read exactly matches, Hamming distance is great, right? You get 8 matches. The problem is your read may have an edit. Basically, there's one deletion in this read. Now it's no longer Istanbul, it's Istanbul. And you want to match this, right? And unfortunately, Hamming distance, if you do this Hamming distance by padding the last one, you get 3 matches and 5 mismatches over here. It really doesn't tell you anything about the similarity of these two strings, right? Sounds very bad. Now the realization in this work is if you actually shift the read a little bit, once to the right, you cancel the effect of that deletion that you have in the read. So you shift, you do this Hamming distance once without shifting. That basically is no edit that you have. And if you shift this to the right, you basically think that, oh, there's some edit over there that I'm not accounting for. And you do this matching, you do this Hamming distance computation again. Now you have 4 matches over here, right? And if you want to account for another edit, you may want to shift it once more time. And account for another edit, you may want to shift it one more time. If you want to account for an insertion, you shift it to the left. So that's the basic idea. By shifting, you're helping similarity search. So now we have 2 Hamming distance results. One is saying I have 3 matches over here. The other is saying I have 4 matches over here. Now what do you do with them? basically, I'm repeating the same thing. We do these Hamming distance computation. Hamming distance is essentially XOR, right? And in the end, you get a bit vector, and you AND those bit vectors. Once you AND those bit vectors, you'll have another bit vector that basically has a 1 on places where you don't have a match in any of those shifted Hamming distance computations. That's the idea. And you count the number of ones. If that is less than a threshold, that tells you that these strings are similar enough for your purpose. This cannot be perfect because there may be different things. I'll show you a pictorial version of this. That's why it's a filter, right? If you want to make this perfect, it's not very easy to actually develop it, but I'd encourage you to try to think about it. So now we have 7 mismatches and 7 matches and 1 mismatch, okay? So how do you account for more edits? Basically, you shift more, right? That's the idea. So pictorially, what we're doing is, instead of doing edit distance computation of these reference and query matrices, what we're doing is shifting. A string is looking at a different part of this matrix, right? So this is, for example, 2 deletion Hamming mask that you have by shifting the query a little bit. And if you do these shifted Hamming distance computations, you don't basically have any dependencies. You can do this completely in parallel. Okay, so we did this completely in parallel and use SIMD instructions. We get good results. But of course, this sort of bitwise operations fits really nicely on FPGAs. And we developed an FPGA architecture called Gatekeeper, which we also published in Bioinformatics afterwards. And we get significant performance improvements. Again, I will not go through this in detail, but it's a full FPGA design, and you can download it and use it. Use the next versions that I'm going to talk about because they're better. And you get significantly faster filters compared to the software filters, best software filters that we had at the time, and we saw significant speedups in end-to-end read mapping at that time. Okay, so the conclusion is FPGA-based pre-alignment, we call this pre-alignment filtering, greatly speeds up read mapping, and we believe that this would enable real-time filtering while you're sequencing, and hopefully paves the way to on-device genome analysis. So there's more on Gatekeeper in the paper. The question is, of course, can you do better? And we optimize these FPGA designs, change the windowing, how far you look at, for example. You don't want to do this on entire reads like 150. If you actually have windows, small pieces of these reads, you can more quickly figure out what may not be matching, and you can actually reduce the computation overheads a lot. And that's the idea in Chuji. Again, I will not go through this in detail, but that's a much better filter. And you can find it online also on Bioinformatics again. I will talk about this one because I think this is very interesting because we were inspired by mapping this problem to some other problem. And this is one of the best filters that we designed. Basically, if you want to compare, these are actually very interesting things that people have looked at. If you want to compare, let's say, a reference genome to the reads, the correct alignment in the end is a sequence of non-overlapping long matches. You just want to figure out what those non-overlapping long matches is across that space or matrix. So our key idea in this case was to reduce this approximate string matching problem to the single net routing problem in VLSI chip layout. So if you think about this, okay, people have tried to solve, you basically have some inputs in VLSI and you need to map it to some outputs. And you need to route these things through a chip. And there have been algorithms developed for it. And we are going to map the problem that I mentioned to this problem. And we actually see significant results. Let me give you the key idea before we go into results. Remember those diagonals that we have? We're going to use those shifted Hamming masks. Main diagonal is essentially, so if you look over here, you have a reference and you have a read. And you basically compare the reference to the read without shifting. And by shifting left 3 times, and by shifting right 3 times. So these are actually that matrices, and you can compute them completely in parallel. The reason we're doing it 3 times is we're assuming that the edit distance that we're going to match is maximum 3. Okay. Now, if you look at this and visualize it and make ones black and zeros white, you can think of this problem as taking the inputs and going to the outputs, basically routing from the input entrance to the exit by minimizing the blockages you have, minimizing the black things that you actually encounter. That's the idea. That's why this maps to the single net routing problem. So you want to actually go the longest white distance possible until you get to a blockage. That's your first error, basically, that you encounter. First difference, first edit distance that you encounter. And then you reduce the edit that you have, and you go to the next one that gives you the longest sequence match, which is that one, as you can see. And you reduce the edit. And then you go to the next longest sequence match, which could be either of these, any of these three. And now you reach the exit. Hopefully this is clear, because all of those zeros and ones are actually our comparisons that we had. And we want to find the longest sequence until we get to a blockage, which is essentially a non-matching part. And essentially this way, you can do these comparisons. Now, the good news is you don't actually build the entire matrix beforehand. You can build it on the fly while you're doing this computation. And this reduces the area on the FPGA or whatever architecture you're designing significantly. And that's why the area is very small, actually, on the FPGA. Okay, so basically, this key insight enabled us to actually accelerate a lot of filters significantly. And I think this is still one of the best filters that we have in the domain. Maybe someone will come up with a much better idea, but that's good. I think that's the beauty of this domain. I wanted to talk about this in a little bit detail because this shows that there is a lot of potential for using new algorithms or mapping existing problems to some other existing algorithms that we know how to solve very well. So that's Sneaky Snake. You can also download it and use it. People have used it. We've also looked at other algorithms. So this is one of the works that we published at Micro in 2020. And we wanted to actually look at different ways of doing string comparison that are more hardware friendly. And in this work, my PhD student, Damla, essentially wanted to design a fast and flexible framework that can accelerate multiple steps. And we started with the BiTAP algorithm. BiTAP algorithm is actually a text search algorithm that was developed in the 1990s. And essentially, it uses very fast and simple bitwise operations to do approximate string matching. So the algorithm itself is basically bitwise. And we wanted to modify it such that we can do very highly parallel approaches. And one of the issues with BiTAP is it doesn't perform traceback, so we need to add traceback. Again, I will not go into the details of this because we don't have a lot of time. But this algorithm fits really nicely the hardware accelerators that you design. And we designed a specialized hardware accelerator for this, which using a modified version of the BiTAP generates bit vectors and performs edit distance calculation. And does traceback to figure out actually which parts in the end match to get the optimal alignment. So the takeaway is, there's a lot of detail clearly over here, but the takeaway is you can build a nice systolic array to do this sort of computation. And I think this is also interesting because people use systolic arrays for matrix computations, but we can also use systolic arrays. It's a very general idea, clearly. It was developed in the 1970s. And we can use these systolic arrays for edit distance computation and genome analyses. I think one question always is, can we actually use these TPUs that are developed for machine learning for better genome processing? What can we remove from them? I believe you can remove a lot from them to actually use them as a simplified systolic array engines. And I think this could be a better pathway for adoption because people are clearly developing these TPUs in a complicated way to do something. But maybe we can use it for genome analysis also. That's kind of a challenge to interesting students over here who want to do great work. Okay, again, I will not go through the hardware design over here. It's relatively simple. We worked with Intel actually to design an ASIC version of this. They were actually quite interested in genomics at the time. And we used their methodologies to estimate area and power. So in the end, the overhead of this is not so bad compared to a Xeon CPU core. Okay, so you can use this for multiple use cases we show in the paper, filtering, read alignment, etc. And we used it for multiple of these. And there could be other use cases also, for example, indexing, whole genome alignment, generic text search. So in the end, what we're doing is text search in a specialized way, but generic text search can benefit from a lot of these algorithms also. And we see significant results compared to best prior software as well as hardware approaches. I'm not going to bore you with a lot of numbers. You can find them in the papers, etc. So after GenAsm, we wanted to actually look at other ways of doing it. I'm going to skip Scrooge, but this is actually generalizing the sequence alignment to CPUs, GPUs, ASICs. You can also put things on FPGAs, but we just didn't do it, didn't have the time. You can look at how we do it in the paper. This is also in bioinformatics. But we wanted to also look at some other problems. Well, one of the interesting things in genomics is algorithms are also changing at the high level. You don't want to design architectures that are actually matching exactly one algorithm. That's why I think reconfigurable computing devices actually are a very good fit, because you can update the hardware that you have nicely. So one of the algorithms that we were really interested in is sequence-to-graph mapping. So normally, what I just described so far is sequence-to-sequence mapping. You have a linear reference, and you have linear reads. But one of the problems with linear references, it doesn't really represent the whole population, right? Some humans are very different, and different humans in different parts of the world are significantly different from each other. So it makes sense to actually have a reference genome or multiple different reference genomes. The question is, how do you actually represent multiple different reference genomes that have some similarity with each other? So people use graphs, essentially. Instead of representing a reference genome as a sequence, you have graphs with some potential probabilities. And now, if you want to check if a read maps to a reference, you need to check all of the possible paths in the graph, right? And this makes the problem more complicated. Right? Now you have a sequence-to-graph mapping problem. And I think in the future, we may want to have graph-to-graph mapping, which is actually even more complicated, but I believe there will be applications to it, even if there are no applications today. So basically, there was no prior hardware design for this. There were software methods. They're also improving. So we wanted to actually design a universal algorithm hardware co-design such that we could extract both sequence-to-graph mapping and sequence-to-sequence mapping for both long and short reads. And we modified our GenAsm design that uses BiTAP. This is the graph mapping pipeline, by the way. You construct the graph genome, and you still need to do indexing so that you can do fast search on the graphs. And this is the full pipeline. There's the sequence-to-graph alignment is the difference that you have from sequence-to-sequence alignment. So this is the hardware design we came up with. Again, I will not go through detail because otherwise I will spend the entire time talking about every single paper. But these are hopefully inspirational in the sense that there are really interesting problems in this field that can be solved with interesting hardware designs. So we basically have a seeding accelerator that finds candidate regions that you want to focus on in this graph, essentially, or it could be a sequence also. And then we use a bit vector-based BiTAP algorithm to do the alignment. So bit vectors are really the key, and I've given you an example of this. In the end, if you design the hardware for this, you get significant speed-ups compared to both software and hardware approaches. And we actually do better than the sequence-to-sequence alignments because we optimize some things better in this work. Okay, there's talk video. This was part of Damla's thesis. She was my last student to graduate at Carnegie Mellon. So she's working in bioinformatics companies right now. Okay. Any questions? I'm going to step back a little bit. And we still have time, I guess, right? 10 more minutes? Yes, one question.
You know that you work a lot with the memory computing.
We're going to get to that, hopefully.
So I was curious about also your take on sequence graph processing, within memory.
Yeah.
So which is kind of a very complex problem, but also highly exciting from my perspective.
Yeah. So you're talking about general graph processing,
Yeah.
like graph analytics, not the sequence to graph. Yeah.
Yes.
I mean, we have a lot of work in that area, actually. I think it's really important, and doing it in memory is actually a very good approach. We can talk about it separately. Yeah. It's a more complicated problem than this one, actually, because it's more generalized. There are a lot of graph algorithms, but we actually have some designs to do algorithms, express algorithms in very similar ways to, using bit vectors, like set approaches. We call the SISA, set-based instruction set architecture for graph processing. It was published in Micro 2021, but I think there's a lot more to do in that area. Yeah, very good. I think it's good to also look at the similarities and differences between graph processing in general and also graph-based genomics. Okay, I'm going to step back over here and foreshadow some of John's talk, but I'm not going to talk a lot because he's going to talk more about this, I believe. I don't know. Maybe he's not going to talk about it, but then you can talk to me afterwards. So let's step back a little bit. So what we do is, in genomics, is you have raw reads. These are some sort of signals. Either you have electrical signals or you take an image. Illumina works. The reason it's called Illumina is it takes images at a fine grain and basically tries to figure out what that is. And then you call bases. This is called base calling. You need to translate these signals to text. Okay. The question is, do you really need to do that translation, right? So we're going to talk about nanopore machines. Nanopore machines are very interesting because it makes a lot of these analyses portable. And the key idea over here is you pass DNA through a nanoscale hole. And while the DNA is going through this nanoscale hole, there's ionic current that passes. And there's a change in current that happens while the DNA is passing through that hole. And the idea is to identify this change. And the change is different based on the bases that pass through. Right? So you basically measure the electrical signals that happen while the DNA is passing. Okay? And there's more information on nanopore sequencing in our overview paper that looks at how to actually analyze it. This is a little bit dated right now, but it's also a good introduction to nanopore sequence analysis and the computational bottlenecks at that time. So the question is, can we actually operate directly on these electrical signals? Right? Why do we have to convert the electrical signals to text? And it turns out this electrical signal to text conversion is extremely expensive. So we do some work in this area. Actually, this is a work that we did with AMD. Gagan, one of my PhD students, spent some time at AMD, used their FPGAs, and we actually accelerate the base calling. And the base calling algorithms that are developed is really based on neural networks. And these are sophisticated neural networks trained using some data that we don't necessarily have access to. But they're also open source models that are a little bit less accurate compared to what nanopore uses. So in the end, you're doing neural network inference to figure out whether an electrical signal maps to ACTG. Right? That's the idea. And that neural network tends to take a lot of time. And here we actually use FPGAs to accelerate that neural network. We develop some techniques. If you're interested, you can take a look at it. I'm not going to talk about that. So one approach is really to accelerate the base calling mechanisms, neural networks using new architectures, new algorithms, etc. And I think that's good to do. The question is, can you do something completely different? And I think I'm quite excited about this. And maybe in the end, we want to combine both approaches, but we should really also examine completely radical approaches that get rid of this sort of base calling. And this is part of what John's PhD thesis was about. I think he's continuing this right now, and there are multiple works that we published in this area. Basically, base calling is accurate, yet costly. Can we actually get rid of it? Can we take these noisy raw signals and directly do read mapping? To be able to do that, you don't want a text-based reference genome. You want a more, let's say, signal-based reference genome, let's say. You want to represent the reference genome using some sort of raw signal convention, electrical signals. And you want to map these noisy raw signals that you have during reads to that reference genome. So this is, John may go through some of these slides. Are you going to go through some of these slides later on? Maybe not. Maybe, okay. I'll accelerate your talk if people are here. Okay, so if the observation is that identical molecules generate similar raw signals. And the key idea is to perform the similarity estimation based on vector similarity search on raw signals. There are multiple approaches you can do this. The question is, how do you do it cheap, right? One way is clearly approximate neighbor search, right? That's one way of doing it, but that may be expensive. So nearest neighbor search, essentially. It's too costly for large genomes. And you can see we did a lot of studies on it. This is in John's thesis. As genome size increases, this becomes even more costly. You may not be able to match the data generation speed if you're actually doing these comparisons using nearest neighbor search methods. So it doesn't enable real-time analysis that we wanted to do initially, right? So what John's insight was, so if you want to do real-time analysis, actually, the idea is you don't wait you don't wait for everything to be sequenced to do the analysis. You actually want to sequence and do the analysis while you're sequencing. And these nanopore devices allow that. And maybe he'll talk about that a little bit more. And also, you can stop sequencing early if something doesn't match. Right? There are methods where you can actually stop the sequencing in a nanopore sequencer. So how do you do this, essentially? So John's insight was to perform similarity estimation based on quick exact matches. And this is a hash-based mechanism. You take a hash of the read, and you take a hash of the reference, and you compare those hashes. And if the hashes are similar enough, you declare a match. If the hashes are different enough, then there's no match. So that's the idea, basically. Then the question is, how do you actually do this hashing? And maybe he'll talk about this in more detail. You need to basically do, actually, a lot of methods that are developed for machine learning are applicable here. You need to do adaptive quantization. And you need to carefully actually form a hash based on that. Okay, so this is the first hash-based search mechanism for raw signals, and we saw significant performance improvements with this. The accuracy doesn't necessarily match base calling, because base calling is very expensive. But the idea is to actually, there has not been a lot of work in this area to develop this sort of methods on raw signals. So we actually want to get the accuracy as much as possible close to base calling without losing speed. And there's still a challenge over here. Okay, I'm going to skip this, but basically this is open source. You can ask more questions to John if you're interested. Do we have more time? Okay, I think yes. Okay. Then I'm going to talk about processing in memory a little bit, because I think that's also important. This is an area that we've been working on separately from bioinformatics, but it actually matches bioinformatics really nicely also. So the idea is basically today's systems are very processor-centric. There's a lot of places where you can process data, and we want to process data everywhere. I think sensors are going to be even more important with the genomic analyses, because that's where you ingest the data. And if you can actually put processing as close as possible to the sensors, do some of the analyses, even faster analyses, do the filtering, for example, nicely, right next to the nanopore sensors, you can probably do a whole lot more. Okay, so but I'm going to talk about processing in memory a little bit, because if you don't do processing in sensors is limited to when you're ingesting the data, right? But we have also a lot of genomic data that's stored in database, and that's increasing. And you want to do analyses on this huge amount of data, then you have to actually do processing in the memory or in the storage. So there are many different use cases for genomics also. So processing in memory is essentially the processor asks memory, can you actually do computation for me? And memory does the computation and sends the results. There are many questions over here that I will not bore you with. I'm giving a talk in the other workshop. I should probably not be advertising for the other workshop, but if you're interested in processing in memory, you can come to that later today. So there are many interesting questions over here. There's also a path for adoption. You will see some real devices where we did a lot of studies on. The question is, how do you actually adapt genome analysis algorithms to fit processing in memory? So genome analysis is actually very data bound. In the end, you see a lot of, if you optimize the computation, eventually you'll become data bound, even if you're not data bound yet. So there are a lot of memory intensive portions of these algorithms. So we basically looked at pre-alignment filtering, and we accelerated. Sneaky Snake was the algorithm that I mentioned, like that takes the problem and modifies it to map to VLSI net routing. And we actually accelerated using FPGAs. This is work Gagan did together with IBM. And the idea is perform read mapping using specialized logic near memory. And we carefully redesigned the accelerated logic of Sneaky Snake to do near memory computation to exploit high bandwidth memory. Again, I will not go into the details, but if you do this, you get significant performance and efficiency improvements. So in this case, what we did was we offloaded computation from a Power 9 CPU to an HBM-based FPGA board, and you have an FPGA right next to the HBM. This is IBM's OpenCAPI method. And we basically accelerated genome analysis as well as weather modeling in this particular work, and you get significant performance improvements. The details are in the paper, but I think there's more to be done in this area, and certainly FPGAs coupled with HBM are important. And going forward, I think, I mean, this is not 3D stacked, but if you have 3D stacked memories, and you have memories stacked on top of a nice logic layer, and the nice logic layer, you can reconfigurable computing engines, that's really a better way of doing it, I think. Of course, you'll be limited by capacity, so you need to think a little bit better. But I think technology is on our side, actually, to do this sort of near memory processing going into the future. Okay, so that's the paper that talks about it. Actually, that was not the first work that we did. We wanted, when we actually looked at filtering, one of my PhD students, Jeremy, who also graduated from CMU, was interested in bioinformatics and mapping some of these filtering algorithms that we developed for processing in memory. And while he was actually trying to map the filtering algorithms to a 3D stacked memory engine, which kind of looks like this, you have a logic layer, he figured out that if you change the representation of the reference genome, you can actually much more easily do these bit vector-based comparisons. So it's important to actually change how you represent the data also. Otherwise, it may be very difficult to take advantage of the parallelism that you have in memory and logic. Again, I don't have slides for this, but this is a work that is very interesting, one of the early works that looks at how do you redesign the representation of the reference genome such that it's much more friendly for processing near memory? Okay, so more recently, we've been looking a lot into storage because data is really exploding in this area, and it's too expensive to store things in memory, basically. Storage, like flash memory, is cheap, and if you actually add processing capability next to the flash memory, you can do much cheaper designs. I will show you some cost analysis also soon. So this is one of the works that Nika, who recently graduated at ETH from my group, developed. It's called GenStore. The idea is basically we bring a lot of data from the storage system. Assume that you have huge reference databases. You bring the entire reference genome to compare to the reads that you have in the computation. So you have computation overhead, you have data movement overhead. People have developed a lot of mechanisms to reduce the computation overhead, including processing in memory techniques. Lots of things which reduce the computation overhead, but the data movement overhead from storage remains and becomes a bigger bottleneck. Whenever you accelerate X prime becomes a bottleneck, basically. So the idea here is to filter the reads inside the storage carefully, such that you move only a little amount of data that really requires the expensive computation units over here. So add some simple computation units to do filtering in the storage system. So how do you do that filtering? It turns out Hamming distance computation is cheap, for example. You can easily figure out exactly matching reads. And if things exactly match, depending on the use case, there may be things that exactly match 80% of the time, right? So you immediately reduce 80% of the data movement from storage to computation with very, very little hardware cost. You can actually do it in the CPUs that you already have inside the storage system today. Non-matching reads, if a read doesn't match the reference genome, let's say very significantly, you can also detect that relatively easily. Not as easily as exactly matching reads, but you can actually add a little bit more complexity to the storage system to do that. You can also detect that. So these are the two accelerators that we added to the system to do the filtering. Again, I will not go through the slide. The paper has a lot of details. And essentially, the key issue to tackle if you want to design an in-storage accelerator is to make sure that your system complexity doesn't explode, basically. You have limited hardware resource inside the storage system. And if you want to do something like this, people don't want to revamp the storage system. I think we should be doing that also, actually, in academia. But if you want things to be more adaptable, you want to modify the storage system as little as possible. And that's one of the challenges over here. You can read the paper for how we handle these challenges. You want to minimize changes to the FTL. You want to also do storage while actually you're doing computation, etc. And also you want to cater to different behaviors that you have in different read mapping workloads. In the end, on average, we see significant performance improvements and also energy reduction at the same time. In the best case, you improve both by about 30x. And the good news over here is you improve both performance and energy. It's kind of like FPGA-based acceleration. You improve performance and energy, right? And that's what we want. Okay, so more recently, this is one of Nika's recent works at ISCA. This is a very complex full system level paper. And the idea is metagenomics acceleration. This is, you have, in metagenomics, you may have databases that are like hundreds of terabytes, essentially. You're not going to use memory for that. If you use memory for that, you're going to pay a lot. So the idea, this is the first in-storage system for end-to-end metagenomic analysis. And the idea is to partition your workload between the host and the storage over here, such that they cooperate with each other. Because there are parts of the workload that are actually really compute intensive. There are parts of the workload very memory intensive. How do you do that partitioning? And I will not go through how you do that partitioning. There's a lot of work that goes into it. And that's, again, a full talk. But I will give you this result because I think this result is really interesting, right? This result compares a cost-optimized system with a not so bad SSD, but not so good SSD either, not high bandwidth SSD, cost-optimized SSD, with only 64 gigabyte host DRAM. This is not in the SSD, this is in the host. So you have a host with 64 gigabyte DRAM and you couple it with a really cost-optimized SSD. We're comparing it and you do computation it. That's what this MEGIS is, cost-optimized SSD with design for metagenomics, essentially. And then we look at a performance optimized system. Host has 1 terabyte of DRAM so that you can actually do this genome analysis with large amounts of data. And we have a performance optimized SSD, very high bandwidth. And if you compare the performance of these, we see that a cost optimized system is much higher performance because it can do computation on the storage side. So I think my takeaway over here is, and it's also much less costly. So even if you have a much less costly system, if you do computation inside the storage, you get much higher performance. And I like this picture because I've always been arguing that our systems are very imbalanced. We actually complicate our systems a lot without gaining a lot of benefit. So if you actually do computation somewhere else, you can actually have a much less costly system in the end, because you don't need to increase the memory capacity, because the memory capacity is somewhere over there in the storage, right? So this enables us to, I think, to rethink how we design our systems. And I like this picture a lot in the end. We have seen this picture for in-memory processing also. You can reduce the size of things. You can get rid of a lot of caches in the CPU, for example, if you actually have very good processing in memory systems. So I think we should be striving for this. And also, in this particular case, we had we used acceleration methods near storage. And we actually have like ASIC type designs. But I believe reconfigurable computing has a lot of role to play, because in the end, we don't want to bake ASIC type designs into a storage system. If you have something reconfigurable and something small, you can actually do a lot in the storage system. Okay, we have also a mechanism to accelerate raw signal analysis. I think Konstantina is going to talk about that, so I'm going to skip this. If you're interested in PIM, we have a lot more, including graph processing that you mentioned. And it's in this 100-page paper that we need to desperately update because it's out of date right now. And we have more papers. Okay, I'm going to skip a lot of these. I will say that if you're interested in PIM, you should come to our memory-centric computing workshops. We are organizing some more at ISCA this year and ICS, and there will be more of these. Do we have time, or are we done? I will mention some real results with real PIM systems, and then I'll conclude. Let me mention this because I think this is interesting. So there are real PIM systems that people are, PIM meaning near memory processing systems. There's a lot of terminology issue. If this was a PIM talk, I will give you the terminology. But you have actually processors next to memory. Samsung, SK Hynix, Alibaba have been designing next to each DRAM bank in a DRAM chip. They're modifying the DRAM chips to accelerate matrix computations. UpMem was the first company who commercialized the system. They were unfortunately bought by Qualcomm recently. Hopefully it's fortunately. Hopefully Qualcomm will do something good with it. But I don't know. I'm not Qualcomm. But basically, we worked a lot with the UpMem company. What they did was next to each DRAM bank, a DDR4 DRAM, they have a multi-threaded processor. And you can program it using simple methods, etc. So you can actually use this to accelerate applications. And a lot of people did, including us. But one of the things that we use this real system, by the way, we have 3 of these servers. If people are interested in doing something interesting with UpMem, we're happy to provide access because UpMem doesn't exist anymore. But we're happy to provide access. Basically, these are the DIMMs that they designed. And these are real. And this is the system that we have. There's PIM-enabled memory. And we have a lot of talks that describe it. So this is actually what you have next to each DRAM bank. You have a processor, a multi-thread processor. I wouldn't be designing things like this. I'm not UpMem, but still, this is a real prototype that you can build real software on. So we did a lot of other things like ML training on this. But one thing that this really shines on is bitwise operations. So you can actually map a lot of the sequence alignment algorithms to this really nicely. This is one work that we did where we mapped the sequence alignment algorithms, and we see significant benefits from processing in memory compared to both CPUs and GPUs. And we improved this later on, and we have another bioinformatics paper that talks about how to do this. If you're interested, you can take a look at these. So real processing in memory systems exist, and you can actually accelerate real alignment algorithms. You can develop new alignment algorithms also, if you're interested. Okay, of course, you can develop new processing in memory systems also to accelerate different parts of the workload, but I'm not going to talk about that because I think we're already kind of out of time, right? Yes? Okay, I see. So we have some gap in the talks. Okay, that's fine. So I will not talk about this, but we actually also improve data movement. So you can see some of the numbers over here. There's a huge amount of data movement that you need to do. And there's a lot of computation. If you can reduce the data movement and also reduce the computation, it's always better. And the techniques are, if you want to reduce the data movement, there are algorithms that you can use. Processing in memory is the hardware support. If you want to reduce computation, you need to actually have an idea of the quality of the things that you have. For example, if you identify that some reads are not high quality, you can actually get rid of computation on those. And this paper talks about some of those that I will not talk about. Let me see. Let me talk about future opportunities, and we can take questions. So I think there is a lot of opportunity going into the future. Like technology is evolving. Like when we started first with the Oxford nanopore technology, its accuracy was 85% or so. Right now, we're much closer to 97% or so. Maybe it's improving. If you talk to Oxford Menopur, they're actually better, perhaps. I don't know. So accuracy of these technologies is improving. So this long reads that you have is becoming better and better. And you can actually do a lot of these analyses in a portable way. Graph genomes are applications that are actually very interesting, I think. I think we need to be thinking about these complex ways of representing genomes, pan-genomes, graph genomes. Another thing that's interesting is if you actually have reference genomes that are frequently updated, how do you actually design mechanisms for it, right? People actually are trying to update the reference genomes as much as possible. If you discover new species, for example, if you discover new variants, you want to have mechanisms such that you can update the reference genomes. So your graph starts exploding, but you don't want to also do computation on parts of the genome that doesn't change, right? So this is proposing one mechanism to reduce unnecessary computation when the graph genome, pieces of the graph genome doesn't change, or piece of the genome doesn't change. It doesn't need to be graph. So we have actually tools for that also. These are part of Jeremy's work. And John actually did a lot of work on that also. So I think raw signal analysis is a new frontier. Like, how do you rethink the system and the entire algorithm so that we get rid of computation as much as possible? So if you, in this talk, I think if the most important thing is getting rid of computation, right? Ideally, we want to not do computation, right? Or focus our computation on the most difficult parts. If we can get rid of unnecessary computation, like mapping to text, I think there's a lot of opportunity over there. The question is, when can we do this accurately, and when can we not do this accurately? And designing a heterogeneous system that can actually partition the workload such that you do the right thing at the right time. So we have works in that area. So there's a bright future, I think, over here. Now I'll conclude. I think the good news is things are happening in industry. We were talking with Madhura yesterday. Certainly, these companies that design sequencers are actually heavily using some of these hardware accelerators. They don't necessarily go public about it. They don't talk about it. But they're actually teams that develop FPGAs, for example, to do some analysis, some acceleration, and some things that we don't know about because they don't talk about it. I wish they were actually more present and they would talk about it. But maybe this sort of workshop would be extended in the future to include some of those folks that are doing the hardware mechanisms in those companies. In the end, I think if they collaborate with academics or people, other people in industry who are willing to actually provide support, this ecosystem can be much more easily developed. I think that's something that machine learning folks did really nicely, right? People who actually design machine learning algorithms collaborate a lot with people who developed hardware for machine learning. I think we have a missing part here in genomics and life sciences. People who design the systems for, let's say, sequencing, etc. There's a gap in terms of collaboration of those folks with academics as well as industry who can actually supply the hardware in a nicer way, let's say. So people have been using GPUs for many things. I'm not going to talk about that. GPUs have been around and they've been used. In fact, NVIDIA has been supporting, for example, dynamic programming instructions that could be useful for genomics, but could be useful for other things also. Yeah, that's the dynamic programming instructions. Okay, let me go back to how we started, basically. So this is what we started in 2006, 2007, right? We wanted to have an embedded device that can do a lot of things. I think we're still a long ways to go. We covered a lot of ground, and I think the numbers of acceleration has been a lot. The problem is we're not there yet. I mean, there are a lot of issues, of course, to enable something. Energy efficiency is really important, performance important, and we're not even talking about security and privacy yet, right? So if you want to do analyses in the cloud, security and privacy immediately become a problem. I think it could be enabled potentially if you actually have privacy preserving mechanisms like homomorphic encryption, different things basically that can enable things on the cloud, but those have actually additional overheads that we didn't even talk about. If you actually do things embedded, you still need to worry about security and privacy because you don't want to lose that embedded device and have all of your data. But it's a less, let's say, space that's actually less open to security and privacy problems. But that's very important also, so we need to think about that. There are a lot of interesting works actually on doing essentially privacy attacks. Someone can figure out a lot of things about you and your family and maybe even bigger circle that you have based on your genomic data, right? Okay. I think in the end, we have a huge memory and storage bottleneck that could solve some of these. Like if this was a processing in memory talk, I would talk about how we would enhance security and privacy using processing in memory also. So this brings me to the end. I think I've given you a lot. a lot of things to think about also. We covered a lot of recent ideas. This is my personal journey, I should say. There's a lot of other work in this area also, right? It's a huge area. So if you're interested in those other works, probably you can start with some of the overview papers that cover not just our works, but also the broader field. But we cannot cover the broader field in a single paper also, right? There's a lot of work. So it's best to go and read and figure out how to read those papers that are not written to be understood. So many future opportunities exist, especially with new technologies and with new applications and use cases. And I don't think there will be an end to, certainly not the application and use cases. Technologies are much harder to develop, like a new sequencing technology, a new nanopore device that's fundamentally better, for example. It's not easy to develop, although there are people who are trying to develop that as well. So I'll conclude. If you're interested, we have genomics courses online. We still offer them, actually. Nika is offering an architecture for health. I will put a plug for our architecture for health workshop. That's also coming at ICS. So we started a synergistic effort, but broader, on architecture for health. The first workshop was held, where did we hold it? Micro in 2025. The next edition was an HPCA, and the next edition will be at ICS. So people who are interested in this topic have a lot of venues to actually discuss, and we actually have a full day at ICS. There are like 10 plus talks from different people talking about different aspects of architectures for health. I'll acknowledge a lot of people who did this work. Some of them are going to talk over here, but now I can take more questions. Before I take this, we actually open source essentially everything on our GitHub. So everything that you heard about is open source online. You can build on top of it more easily. And I think that's one of the nice things in bioinformatics. I kind of criticize bioinformatics saying that it's written to be not understood, but they also open source a lot of the code, which is really good, actually. And I think that more of that should be done in other communities too. So that brings me to the end. I'm happy to take more questions.
Great.
Thank you. I have a very basic question for you. How do you get these reference genomes in the first place?
Oh,
Because it's a chicken and the egg problem if you just consider what you presented, if you are from outside of the field.
So the good news is these reference genomes exist. For human genome, for example, there was a large human genome project. How do you get them in the first place? Basically, you need wet lab experiments. You need to have a wet lab. You need to actually figure out what the genome looks like. You need to reconstruct everything. You cannot do it computationally. You can do part of it computationally if you actually do some de novo assembly. The accuracy of de novo assembly, basically, it's a graph-based analysis where you try to, you basically sequence a genome and you try to construct a reference genome without a reference. You still use some references to try to figure that out. It's computational, it's purely computational. The problem is it's very hard to verify the accuracy of it without wet lab. So you need to have a wet lab to actually get the reference genomes. And people actually invested a lot of money for the Human Genome Project when people were more sane and investing in science, let's say, in the entire world. But hopefully that'll continue going into the future. Yeah.
Thank you.
Sure.
So as someone with a more FPGA background and part of the FPGA community, it seems like there's a lot of really interesting ideas to experiment on the FPGA and to also accelerate. So I'm wondering, is the limitation here, like, from someone who's works with all these FPGA tools, is there something in the community that we can do to make the hardware design tools better to prototype these kind of things? Is it just, you hire more grad students to just work with Vivado and Cadence? It's not like all these tools, or is there actually things that we can do at this level to improve the prototyping of these systems?
Yeah. I mean, that's a good question, basically. I think that's a broader question. How do you make hardware design more accessible?
I work with HLS, and this is a big promise that I don't know has been fulfilled, especially for like much more complicated applications like this.
Yeah, basically, I don't have a very good answer, but I think in the end, if you want to optimize hardware, you need to know hardware, right? In that sense, people need to be trained more in hardware. That's why I think we need to have more education in hardware design. And maybe software will be easier to design by the AI agents going into the future, but hardware is not going to be that easy to design, I think, going forward. But I think efforts like HLS can help. Like if you actually have some libraries that are easier to instantiate and people can actually more easily plug and play with libraries. So if I want to do, for example, some read mapping module, and I can have a library of different filtering algorithms, and they can be instantiated on FPGA without me knowing about an FPGA. That would be useful from the perspective of a bioinformatics person, right? From my perspective, I think having libraries is very useful, and hopefully someone optimizes those libraries, right? And not the bioinformatics people.
Yeah.
Because they don't optimize even their software. You will see that if you actually look at the software. There are people who actually optimize software, like Hang Lee. Minimap 2, for example, is very popular because he actually did a lot of optimizations of the software and he keeps optimizing. And those are the software that actually gets adopted in the end, but nobody understands how, nobody meaning, nobody who uses that software at the higher level really want to look at that software. So I think we need to take a similar approach. You have a library that's seamlessly integrated with existing software, and some really smart hardware designer actually designs those libraries, but there's a nice interface such that other people don't need to think about it.
Thank you. Do they know about HLS, these bioinformatics people?
You can ask them and figure it out, but probably not. That's my guess, you know. There are some people, of course, like a small fraction of the bioinformatics community who is really interested, but that's not the majority for sure.
So there is another question here.
Thank you. I have a very specific problem. Could you please turn back to page 100 or 16?
160. Oh wow.
Okay.
Okay, you remember well. Let's see how fast we can go. See if I had an index, I could go maybe 160.
16. 16.
16. Okay. We'll need
Maybe
to
you
go
can access.
faster. Getting there.
Oh yeah, almost.
Are you sure? 16.
16. Yeah.
Okay.
Oh, yeah.
This one.
Yeah, that's
Yeah.
fine. I think one great feature of hash function is that if the raw signal just change a little bit, the hashed value is changed greatly. And in this slide, I didn't get it. I'm not sure if I have missed something. But the raw signal here is a little bit different. But I don't know how can you get the same hashed value.
So basically, you need to quantize things, right? If the raw signal changes little, quantization takes care of it such that the hash doesn't change.
Okay,
So it's not a hash that's extremely sensitive to every precision level. If you quantize things, then and you need to do this, like this, the next slide, adaptively quantized hash value, then you actually are not sensitive to those changes.
Oh, so the quantized raw signal are similar, are exactly the same.
Exactly.
Okay,
exactly.
okay,
Yeah.
yes,
And
thank
John can tell you more if you're interested in more. Yeah.
you. Thanks.
Sure.
So, any more questions? Okay, good. That's good timing, I think. Then I think we can go on with a coffee break and continue with our schedule in 15 to 20 minutes.
Okay, thank you. Did you drink your coffee? Okay, good. Yeah, that's good. You're welcome. Okay, cool. So.
You just need to wear this somewhere
Okay.
close.
Yep.
It's not for amplifying your voice, but it's for to capture.
Understood. So I should speak loud. Yeah.
Do you need this view or do you want to see the same?
This is fine. Yeah.
That's fine.
And do you want me to stand on the podium or anywhere?
Go ahead.
So camera, there's no issue, right? Okay.
Yeah.
Oh, cool.
I will,
Awesome.
only-
Awesome.
Help them because now, sorry for the.
Oh, it's okay.
There was one,
Is it still 25?
yes, it's the same
Okay.
because actually one of our speakers didn't show up.
Oh, okay. Perfect.
So we're back on schedule.
Okay.
I just thought we could extend the line.
Yeah. Makes sense. Yeah.
Okay. So everyone, I think we're getting started again. So let's continue with our schedule. So I'm very happy to introduce Yetis. Yetis is an assistant professor in electrical and computer engineering at UC San Diego, with affiliations in the Department of Computer Science and Engineering and the Bioinformatics and Systems Biology graduate program. He's a recipient of the MIT Technology Review's Innovators Under 35 Award, the Hellman Fellowship, the Jacobs Early Career Award, the Amazon Research Award, the NVIDIA Graduate Fellowship, and multiple paper awards. So Yetis, you can go on.
Thanks a lot, Konstantina, for the kind introduction. And it's great to be here. Like Onur, I also had a conflict last time this workshop was held, but I'm very happy that this is the second edition of it, and I got to participate this year. Today, I'm going to be talking about nearly a decade of experience that we've had in accelerating bioinformatics on FPGAs. In my lab, actually, we work on a broad variety of bioinformatics problems as well as platforms. Like we use GPUs, we use a lot of high performance computing. But today's talk is going to be primarily focused on accelerating bioinformatics on FPGAs, which we find to be very exciting and promising. So much of bioinformatics is actually about analyzing biological sequences. These include DNA, RNA, or proteins. And these are essentially like these biomolecules that encode the function for that encode the information for the functioning, growth, and reproduction of life. And the interesting thing about all these sequences is that they are linear sequences composed of different alphabets. For example, DNA is composed of alphabet A, T, G, C, so 4 letters. And similarly, RNA also has 4 letters, where the T is replaced by U. And proteins have an alphabet of 20 letters called the amino acids. And like Onur had mentioned, that with the advent of DNA sequencing technologies, we've actually seen an exponential growth in the data of all these different types of sequences. So this is a plot that I'm showing for released by NVIDIA, which is showing how the growth of DNA sequence data has occurred over time. And as you can see, we actually have seen this exponential rise, but also an exponential decrease in the cost shown on the gray curve. And a similar trend can be observed for other types of biological sequences. This includes RNA and proteins, which have also seen an exponential curve, as you see on the right. This, of course, has a y-axis on the logarithmic scale. Now, what's interesting is recently a San Diegan company, a startup, Element Biosciences, has unveiled a DNA sequencer which can sequence the entirety of the human genome in only $100. What that means is that the cost of DNA sequencing has decreased by 1 million-fold in about 25 years time period, right, which is far outpacing Moore's law. And what that also means is that the bottleneck in DNA analysis and decoding the function of the DNA and other biomolecular sequences has shifted from data generation to data analysis or bioinformatics. And the sequencing behemoth, as Onur had also mentioned, is Illumina. It is also based in San Diego. And a lot of the FPGA community is actually not aware, but they have this product called the BioIT platform, which they use for accelerating the data analysis for DR sequencing data, which is actually very widely used. A lot of bioinformaticians who use it are also not aware because this platform is actually deployed on the cloud and gets used for many different types of analysis that they support. Now, there is one thing to generate biological sequence data, which of course DNA sequencing technology has enabled at scale, but individually none of these sequences would make sense. When they make sense is when you start comparing these sequences, right? So comparison of biological sequences is extremely important. For example, you might have collected 4 different sequences like this from different organisms. And when you compare these sequences, that's when you realize, for example, that the sequence S1 is more closely related to S2 than it might be to S3 or S4, right? So you can actually build a phylogenetic tree structure, like the evolutionary structure that I have shown over here, based on the comparison of these sequences. And this has many different applications. For example, it can be used for reconstructing the ancestries of different human genomes, or it can be used for building the evolutionary trees, such as the species trees for the different forms of life that we have. And similarly, sequence comparison has applications in what is referred to as homology detection. What that means is once we have sequenced the entireties of the genome for different organisms, for example, we might have sequenced a mouse and a human, and what this plot is showing is essentially these different chromosomes of these 2 species laid out, where the same colors basically means that that particular segment has evolutionary links back in time between these 2 species. So it sort of shows the correspondence of characters in the genome sequence. And again, Onur Mutlu had mentioned that this type of comparison is also used for the problem of genome assembly, where you have large fragments of DNA which are being read out through these sequencing instruments. And we have to piece together these small fragments to reconstruct the entirety of the genome sequence. And that process is referred to as assembly. Another application which was also mentioned earlier was about genome sequencing analysis, where you have outputs of these sequencing machines, and they actually don't directly give you the sequence of characters like ACGT. Rather, what they give you is electrical signal output. For example, the one which I've shown on the right. It can also be an image output, but for this particular sequencing instrument, it's actually a signal output, and there are going to be talks about this later as well. But that also requires sequence comparisons to decode from going from a signal data to an actual genome sequence data. So all of these comparisons are actually critical for different types of biological analysis. And the paradigm that we use for performing these comparisons is referred to as sequence alignment. And an example of that I have shown on the right figure. Essentially, the goal of the sequence alignment problem is to compare these 2 sequences and arrange them in a way that you're typically maximizing for the number of matches that you have in characters, you're minimizing the number of mismatches or substitutions, and you're also minimizing the number of gaps that you have to insert in the alignment, which would overall maximize a certain objective function referred to as the alignment score. Now, what I showed you earlier was just aligning a pair of sequences, just 2 sequences. But another problem that's super important in bioinformatics is to align multiple sequences, and that is referred to as the problem of multiple sequence alignment. Example of which is was shown on the right. And this problem has actually found applications even in structural biology. For example, Google DeepMind's AlphaFold, which won the Nobel Prize a couple of years back, relies on this particular kernel, as you see in this particular box, that it actually depends on alignment of multiple protein sequences before it actually uses the neural network to perform the structure predictions. So that is about sequence alignment problem. Now I will describe you the exact underlying algorithm behind performing sequence alignment, right? So let's just say that you are given these two input sequences. Typically, what we do is we use dynamic programming, as was mentioned earlier, for comparing these sequences. And that includes 3 different steps. The first would be a step like initialization, where the idea is that you have a dynamic programming matrix, a 2D matrix, in which one sequence appears on the columns of the matrix and one appears on the rows of the matrix. And what you do is initialize the scores of the first row and the first column through a certain formula. And that formula can change, but one such formula has been shown over here, which only depends on certain gap penalty D. Then what you do is you fill out the scores for the rest of the dynamic programming matrix. And that is typically done using a recursive equation like the one which I've shown on the bottom right over there. And depending on which of those equations yield the best score, you actually compute a pointer, which have also been shown for at the different cells, as in from which cell was that particular score derived. And that helps you to trace back the final path or find the optimal path that led to the best score across the whole of the dynamic programming matrix. And it can also help you to recreate the alignment output like the one I've shown on the right. And the most popular such algorithm, dynamic programming algorithm, is referred to as Needleman-Wunsch algorithm. And we refer to the problem that it solves as global alignment problem. And the reason is because it is aligning entirety of one sequence with the entirety of the second sequence, right? When it computes the final alignment. So that's Needleman-Wunsch as shown here. And this particular dynamic programming algorithm has been very well studied, even from the point of view of hardware acceleration. Actually, back in 70s and 80s, people realized that this type of algorithms map really well to 1D systolic array architecture, as was also mentioned, where each processing element at a time is computing one cell of the dynamic programming matrix, and you're exploiting what is referred to as the anti-diagonal parallelism available in the computation of the matrix. So I won't get into the exact computational details of this, but this problem has been well studied in the past, and it maps really well to 1D systolic arrays. And people have also realized that FPGAs provide an excellent substrate for mapping these types of algorithms, because 1D systolic array is very easy to implement on FPGAs. But more importantly, oftentimes the operations involved are just some bunch of comparisons and additions and so on, right? So simple operations, easy to map to FPGAs, and as a result, extract out of high efficiency. So while this was very well known, what was not very well known to the community at the time that we wrote this paper, Darwin, was how do we align really long sequences on hardware like FPGAs, for example, right? And the problem that you encounter is that for long sequences, the size of the dynamic programming matrix grows quadratically, right? Because each of the sequences is long, and you have a 2D matrix in place. And because of the memory requirement, what that means is you can't do the whole computation using on-chip memories. So you have to rely on off-chip memories to be able to do this. And at that point, it defeats the purpose of acceleration, because the overhead is so high that it's orders of magnitude slower to access those memories, and the speed up is negligible compared to software. So what we did with Darwin was to actually map this problem onto a memory limited device like an FPGA by using what is referred to as a software tiling approach. That's where we pioneered. And as you can see here, that it accelerated the problem of long read genome assembly at that time. And we were much ahead of the curve, because at that point, most of the computer architecture community was actually focused on doing short read mapping, which is much easier because it actually fits onto the FPGA memory. But this problem does not. And this particular paper actually won the Best Paper Award at ASPLOS in 2018, and was also chosen as a micro top picks that year. And had worked later that year with Microsoft to actually port Darwin onto their Azure cloud platform. And this is sort of their press release that they made at that time. Of course, since then, the error rates in these long read technologies have dropped dramatically. And so the computational structures that we now require is a little bit different from what we had originally with Darwin. But the problem of, or the solution of tiling is still very relevant. Anyway, post-Darwin, we actually decided to summarize a lot of what we had learned about accelerating dynamic programming, especially in the context of bioinformatics, In this particular paper, domain-specific hardware accelerators, it actually discusses in a broad-based sense, not only for genomics, but other applications as well. It actually appeared on the cover of the CACM magazine. And as you can see from the authors, it includes me, but also my PhD advisor, Bill Daly, who's also the NVIDIA's chief scientist. And one thing that we did was to study how, for example, FPGAs and GPUs, how efficient they are for different domains. And two different domains that we looked into were deep learning and genomics. And what you can see that's pretty interesting in this plot is that for deep learning, for example, you can clearly see that GPUs, even at that time, had a huge advantage over FPGAs. So GPUs are using the green bar and FPGAs on the red. And you can see there is a huge benefit of using GPUs for deep learning as opposed to FPGAs because they were highly optimized for multiply accumulate type operations and linear algebra operations. But what we observed for genomics is that FPGAs actually had better energy efficiency, much better energy efficiency at that time. And of course, Bill, being the chief scientist of NVIDIA, did not like to see this disadvantage. So we started thinking about what could GPUs do to close this gap with FPGAs. And so we actually, in the same paper, proposed an idea to be able to do this, which is to include a special instruction on the GPUs called DPS, Dynamic Programming Step Instruction. And interestingly, a couple of years later, NVIDIA actually launched a product which featured this instruction, which was also mentioned in the earlier talk, called DPX instruction. And the idea is to have a single complicated instruction, like a tensor core instruction for deep learning, which supports dynamic programming problems. And as you can see from their white paper, it actually improved the performance of bioinformatics workloads on GPUs by several folds. So what we saw earlier was the 2D dynamic programming paradigm using and performing global alignment. But that's not the only type of problem where dynamic programming is used in bioinformatics. In fact, the variation of dynamic programming problems is actually very broad in bioinformatics. So for example, if you just modify the global alignment's initialization function, as well as some changes to the scoring function, for example, I'm including an extra 0 in the dynamic programming equation. And if I change how I do the traceback, where instead of starting from the rightmost or the right bottom cell, I'm actually starting from the cell which has the maximum score and I can end anywhere, I actually change the problem and get local alignments. What that means is I'm finding shorter subsegments which align really well with each other. This is referred to as the Smith-Waterman problem. But that's not the only variation, by the way. You could also do semi-global alignment. And again, what you're changing is some of these, some parts of these 3 steps, or you could do overlap alignment. And the differences are highlighted in these yellow boxes. You are changing something in initialization, something in scoring, something in how you do the traceback. So again, lots of variety, right? And this is not it. If you look at 2D dynamic programming, there's other type of variations. For example, I showed you earlier DNA sequences being aligned. You could replace this by the alphabet you can replace by amino acids, which are 20, not 4, right? And then you can perform protein sequence alignment, right? But this changes the hardware requirements dramatically. Or, as was mentioned earlier, that you could do raw signal analysis for base calling like applications, where you could have floating point numbers, but you could also have complex floating point numbers, a series of them. And you can perform alignment for them for base calling applications. So lots of variety even in alphabets. What makes it even more complicated is for multiple sequence alignment problem, sometimes you use these profiles, which means that every letter is not just a single character, it's actually a vector of numbers, which shows the frequency of different characters, right? So every position is a frequency of different characters rather than being a single character. And you can align 2 profiles to form multiple sequence alignments. So lots of variety even in sequence alphabet. Similarly, there's a lot of variety even in scoring. So I just showed you the simple Needleman one earlier, but you can actually change the gap penalty function. And you can get, instead of 1 matrix being computed, you can get 3 matrices being computed for this problem. But they all use dynamic programming again, and it's all 2D based. Or you could replace it by hidden Markov models. And now you can perform Viterbi algorithm, which is also used in bioinformatics. Or you can do raw signal analysis using a single matrix, which is a lot more simpler than what you had with global alignment, but it uses different alphabets again, right? So the point I'm trying to make is, even within 2D dynamic programming, which a lot of bioinformaticians focus on, there's a huge variety, and depending on which application you're trying to solve, you will see a different version of the dynamic programming being used, right? And what's been the general solution for most architects is that they pick one problem or one application and they write the RTL code for it using system Verilog or Verilog or VHDL, right? Spend months of time doing it, accelerate it, get huge speedups, very high efficiencies, but that's not going to be very programmable. So it's not going to adapt to other applications in the same domain. So another possibility, and so I'll just show you some examples of this, and this includes some of my papers as well. But many hardware, like computer architecture papers, have actually adopted this approach of writing custom RTL code for certain specific application and certain specific dynamic programming problem. Examples shown here, one accelerating Smith-Waterman, another Smith-Waterman with tiling, that was my work. And then we used, extended it to Darwin WGA, which does X-drop alignment with tiling. That's one solution, right? But no programmability here. So another option would be, why not use high-level synthesis here? Because that would be very quick to adapt from one application to the other, right? That is what we would generally think, and that was a question that was earlier raised as well, and I'm glad that was asked. And we tried it. We tried this approach, and what we found is our first attempt was actually very quick to prototype, right? Very quick to prototype. Within days, we had a working solution of a dynamic programming problem, except the efficiency was really poor on our first attempt, right? And that anyone who has worked with HLS tools knows this is very, very normal, because you've still not figured out what compiler directives work for this particular set of workloads, right? So what we did is we actually spent weeks of effort trying to optimize it. And we actually ended up closing to a large extent the efficiency gap that we had with the custom RTL. But now it's no longer that productive for us to use HLS, it seems, right? Because it took us roughly the same amount of time that my students would have required to write a custom RTL kernel. Except there is actually hope. And I'll tell you about that hope later. But again, the productivity was not super high. And the advantage that it actually offers is that compared to RTL, which is very hard to modify, the HLS program that we used was actually much easier to modify, right? So if I change the DNA sequence characters to proteins, it's just writing a couple of lines of code, and now I get a protein sequence kernel out, and that's the real insight that we had, that now that we figured out the optimizations, we could actually generalize it to other sets of problems in dynamic programming, right? So the gap that we are trying to fill is the one shown on the black box in this, or the shaded box in this trade-off curve, that we want to have very high efficiency of these kernels on FPGAs, but we also want to maintain programmability and productivity for end users, right? So that led us to developing this framework called DPHLS. And the key insight that it is based on is what I showed you in earlier slides, that there's a wide variety of dynamic programming algorithms used in bioinformatics, right? And they all efficiently map to only one type of architecture, that is 1D systolic arrays, which is what I also discussed earlier, right? But what changes is what function this systolic arrays perform, or what alphabets they use, and so on and so forth. So this particular work was actually led by my PhD student Anshu Gupta and two undergraduates at that time, Yinki and Jason, and was published in HPCA earlier this year. And it is, the artifacts are fully released. As you can see, it's actually released and evaluated as well as reproduced, and is available on this particular GitHub page. It's also portable on AWS. Actually, we've already done that. But what I should mention is that the key idea is that it's introducing the idea is that it's a framework which introduces a layer of abstraction in the HLS flow. What do I mean by that? This framework is meant to empower broad bioinformaticians who are generally software developers and not hardware designers. And what we want to do is that those bioinformaticians should be able to write FPGA kernels without having to have hardware expertise. That's the problem that we are trying to solve, but for this narrow domain of 2D dynamic programming. So what we do is we have a front end and a back end. And in the front end, we only require users to change code in C++ without having to worry about all the hardware design implications that we'll have. And on the back end, we want to define certain optimizations that will occur on that particular specification such that it maps really well to 2D systolic arrays. That's basically what we try to achieve with this particular framework. I should also mention, unlike other HLS works, which are based on libraries that they implement a fixed number of kernels, we wanted to create something that is extensible easily, where you can write a C++ function or modify the code directly and get desired outputs, okay? So that's basically what we wanted to do. Yeah, so that's the main idea. And I'll speed up a little bit now. But the goal is that in the front end, there's a front end and there's a back end. Front end is where the users come in, and back end is something we've implemented on our own. Okay, so front end, what do we allow users to change? They can, for example, change the initialization function, and literally it is going into the C++ code and modifying the function. You can write the function which I've shown here with those 2 lines, but you can create a much more complicated initialization function if you like. Which could be dependent on several other parameters, right? Similarly, we allow users to define custom data types. For example, your alphabet in the sequence alignment could be completely custom. You could have a vector, if you like, a floating point, or you could have a single character, which is only 2-bit of information for DNA and RNA, or 5-bit of information for amino acid. You could do all of these things. You could also have arbitrary number of scoring parameters. You could also have varying traceback start and end pointers if you want. You can customize the function itself using those parameters. You could specify your traceback strategy, whether you want to do global, local, semi-global, anything, right? You can specify that with simple few lines of code. And you can specify what type of parallelism you want to exploit, like how many processing elements on a systolic array versus how many systolic arrays you want to create, inner and outer parallelism. That's the only hardware detail that end user has to worry about, not the internal specifics. That's pretty much it. And then you write the host side program and get your kernel deployed. This is to empower end-bound informaticians, okay? And on the back end, we've done a lot of work, which I can't get into much details, but you can read those details in the paper. But there are optimizations in place so that we exploit the wavefront parallelism that's available in these kernels. It will generate 1D systolic arrays through the hints that we provide. This took the most time, which is to create these local buffers, which are partitioned for doing parallel accesses and making sure that the performance is high and II is low, effectively 1. And then it also performs memory address coalescing, again, something that took us time to actually implement and get it right. Access independent memory banks, lots of optimizations in place. But the point is, at the end of this, we will be able to map really well to the desired hardware patterns, right? So what are the contributions? Essentially, we actually demonstrated that this framework is versatile for very wide variety of 2D-DP kernels. And here I've shown a list of the kernels we implemented on our own. So we implemented 15. And I can tell you that for most kernels, we just spend like a couple of days, 3 days to take it from specification and fully deploy it actually on an FPGA. So after the first few weeks of effort for the initial kernel and developing the framework, which took us time, new kernels were really easy to add. As far as the performance to custom RTL is concerned, we were comparable to custom RTL, although custom RTL still has a little bit of efficiency advantage. If you want to see the numbers, here they are. The throughput is slightly lower than custom RTL, expected. And you can get through HLS even higher performance if you want, but that will require to add more hints and get into specific weeds. But this should be good enough because it's still a huge advantage over software, as you will see, that compared to CPU baselines, you still get about 41-fold higher efficiency on FPGAs without having to modify anything on the back end. And as far as comparison to GPU is concerned, which would be interesting, so I'm comparing to NVIDIA Hopper GPU, which introduced the DPX instruction, which I talked about earlier. And what we observe is that the energy efficiency advantage of FPGA is still there. We see about 2.3-fold more energy efficiency, but GPUs have a lot more functional units, and they consume a lot more power as well, right? But with this amount of parallel processing capability, their throughput is actually much higher because FPGAs, you just can't fit the same amount of logic. So they are about 11-fold higher in net throughput, but lower in energy efficiency. So there's still hope for FPGAs, in my opinion, based on this result. And by the way, here I'm comparing an FPGA that's 14 nanometer technology, whereas the Hopper GPU is 5 nanometers. So three generations difference, right? So that's also important difference to note. So that brings me to the end of the talk, but I'll just summarize some key takeaways that we've learned in our journey so far. First, FPGAs do offer efficiency advantage for many bioinformatic workloads, and that is a very positive news for this community, including over GPUs, by the way. And as you know, that there are fewer and fewer applications where FPGAs hold this type of an advantage, but bioinformatics is definitely one of them, in my opinion. But the problem is that unlike GPUs, which have this moat of CUDA, where developers are really empowered through this type of high-level specification, FPGA design productivity remains low, and the learning curve is just too high. which is why bioinformaticians are today adopting GPUs at a massive scale, but we don't see the same level of adoption for FPGAs unless Illumina-like company just provides you a full product and you just don't have to worry about it. But typically, people don't develop their software on FPGAs. And this is not addressed, in my opinion, with HLS. HLS is what DPHLS is doing is it's actually solving this problem to some extent for a narrow class of applications by introducing a layer of abstraction so that broad applications can be mapped efficiently to the same hardware primitives. But there are some other open challenges. We don't have a good abstraction level available for a good balance of FPGA design productivity and efficiency for applications beyond the one I demonstrated. So we will have to have more DPHLS-like frameworks to be able to do something like this, which empowers people to actually have much higher levels of adaptability and not just fixed libraries. And one disadvantage that FPGAs have is that they're not becoming domain specialized rapidly. But GPUs are doing this very efficiently these days. You already know about tensor cores, which are used in AIs. These are specializations for AI that they have hardened logic on GPUs, which make them much more efficient. DPX is one such example for genomic workloads, but FPGAs haven't done something similar to this. And I feel this is something that the FPGA community should think more deeply about. With that, I'm happy to end this talk and take questions.
Thank you very much. So any questions?
Thank you for the presentation. I feel somewhat vindicated because we had the same issue when we were doing graph neural network accelerators, and we took the same exact approach. So we built this kind of domain-specific compiler, as you might see it. But do you think, this is the way to go for bioinformatics? If we do we just build more of these kind of domain-specific kind of compilers on top of HLS in the meantime while we wait for the vendors to catch up, or are there other approaches that maybe we should also consider?
I'm glad you asked this question.
Yeah.
Because this is actually a common confusion with DPHLS.
Okay.
DPHLS is not actually a domain-specific compiler.
Okay.
DPHLS is a HLS framework built on top of existing HLS compilers. So we use the AMD Vitis HLS infrastructure.
Yeah.
And all we did is add a level of abstraction to it, right? By having our own backend optimizations. And this was purposefully done. We didn't want to create our own compiler because then the adoption is even more challenging, right? We wanted to empower end users today. And HLS has done a lot of work. And you can make good use of HLS compilers if you know how to, right? And still create levels of abstraction on top of it so that productivity improves. And I think that might also be a direction which the community should consider rather than having a new compiler proposed for every new domain. Can we use the existing infrastructure, compiler infrastructure, and add layers of abstraction and improve the abstraction levels that are available?
Thank you.
Yeah.
Thanks for the talk. I have a question more about the architecture and the scale up of these things. Do you think it's possible to try scaling up to multiple FPGA without dying from the off-chip transfers? Because it's a pretty, every symbol is related to every other symbol, so that's pretty hard. But did you consider this?
Very good question again. So in most of these bioinformatics workloads, actually it's so embarrassingly parallel, right? That there is intra-parallelism available, like the wavefront parallelism, which is comparing two sequences, you have parallelism there, which is what systolic arrays exploit. But there is also inter-alignment parallelism. What that means is typically, as if you notice in the Onur's talk, this was also mentioned, that you have millions of these sequences that need to be compared, right? So you have many of these independent comparisons that take place, and that can be independently mapped, right? You can actually partition and independently map it, and very easy to do that.
Okay.
Yeah.
So soon I can
That's how multi-FPGAs can be exploited.
Okay.
Yeah.
So in 1 year I can read a paper about that from your group, maybe?
Yeah, so Darwin is a good example of that paper, and DPHLS too.
Okay.
Yeah.
Thank you so much. So I think we can go on with our next talk to keep on track. And if there are many questions, I'm sure maybe there is some time afterwards. Thank you very much.
Thanks.
So maybe you can wear this, it's a clip-on thing.
too loud.
Okay. Start. So how does this start the?
I'll take care of it. Yeah.
But I have to share it with Zoom. Play.
Yeah. Okay.
Play. I guess it's better.
Oh, this is not the first slide.
Oh, I have to click on it.
Yeah.
But why? Let me share them first because. Where did it go?
Yeah, looks great. Okay, you got it.
I'm sorry.
No,
Right, you have
thank you.
here.
Okay, great. Okay, so yeah, my name is Amir Ali Aghazari. I'm a assistant prof in School of Electrical Computer Engineering at Georgia Tech. We don't work on hardware. We are on the software side. In this talk, I'm going to talk about some of the algorithmic approaches that we have been doing at the intersection of AI and biology, mostly to understand what AI models in biology do. Let's see. Oh, just click on it, right?
Use the clicker now.
Yeah. Oh, okay. So yeah. So like intersection of AI biology has been super exciting during the past few years. What we see is that AI models that are trained on raw protein sequence data can now predict the structure of many proteins. They can predict what is the effect of mutations. Like, is the mutation going to increase the fitness of the protein? Is it going to make the protein more pathogenic, less pathogenic, or function better? I mean, everybody knows about AlphaFold. You can predict how proteins interact with each other. You can also generate new sequences that are completely new to nature. But despite all this great stuff that is happening, we don't really know how these models work. And since we don't know how these models work, we cannot also efficiently and effectively work with these models. And this is going to be problematic in the long run, because if we are using AI systems that we don't really know how they work, then if we want to use them for scientific discovery purposes, then it could be that we are wasting our resources. We could use hypotheses that are generated by AI, but these ones might not be exactly mechanistic. We don't know why they work. And if you think about the history of science, so when we developed these new technologies, from telescopes to microscopes to sequencing machines, we always knew how they work. And of course, they changed the face of science because they allowed us to, in the case of maybe telescopes, see the far objects better. In the case of microscope, we were able to see close objects better. And same thing for sequencing machine, we are able to see what is inside the biological systems. And AI is a technology that helps us to understand what's going on in large amount of data and reason about it. But the problem is that for the first time, we are developing a technology which we don't know how it works. So for all the rest of the cases, we had the idea of how the sequencing machine works, how the telescope works. And that is going to be something that has been always puzzled by, right? So how can we develop this system so we don't fully understand how they work, even though they are predicting pretty well, right? So what we do in our lab is that we develop like algorithmic ideas, mathematical algorithms that are inspired by hashing, streaming, signal processing, and coding theory to be able to understand AI models better. We also develop this sort of mechanistic tools to be able to unravel AI systems, to see what is the computational circuitries that goes on in AI models. We also kind of exploit AI to be able to explain itself. So we developed this agentic system that can help us to understand what's going on in AI. And really, what makes explaining AI hard, it's exactly what makes it interesting. So they are really hard to explain because they're so large. It's the scaling problem that we have to address. So some of the models that I was just talking about, like this ESM model, which is a protein language model, now it's at around 100 billion parameters. AlphaFold is slightly lower, but all of them are around billion parameters. And when we are dealing with these models, even doing one query to them is going to be very costly, right? So let alone we want to query them thousands of times. So for that reason, we need some sort of algorithms that are very sample efficient. So we will be able to interact with them in a very sample efficient way. So in biology, we started with a very simple question and very fundamental, is that can we understand how AI models predict sequence from function or function from sequence? So in this case, let's assume that you have a function f. This can be a massively large AI model, doesn't really matter. You have some input sequence. You start playing around. This is a protein sequence. In this case, could be genome. Doesn't really matter. You start mutating, and as a result of that, the property of the protein is changing. And a really important question, statistical genetic, is that what are the nonlinear interaction that goes on between these mutations? Given having access to F, how can I say what are these higher order interaction? What is the usual way to solve this problem? You need to query F exponentially many times, essentially for all the possible sequences 2 to the power many n times or 20 to the power n many times. And then you need to do something like a fast FFT, which in this case will be a fast Walsh-Hadamard transform because it's discrete data. But you can easily see that this is going to be very time consuming. You cannot query a function f exponentially many times. So what we observe is that these sequences are highly structured, and this structure show itself by sparsity in these interactions and specifically low orderedness. And we were able to slash this q to the power n or 2 to the power n bottleneck to k log n. So you can essentially query these models with a computational complexity that grows linearly with the number of mutations, and you will be able to explain these models very efficiently. And not only that, we saw that you can develop even better neural networks that by inducing this sparsity and low orderness as an inductive bias. We saw that there is an intimate connection between the Fourier transform of an AI model to its Shapley interaction. Shapley is a concept from game theory that was brought to machine learning to help us understand what is the contribution of each feature. So in this case, if you have 3 mutation A, B, C, if let's say this mutation increases the fitness value by so much, if you ask what is the contribution of A, what you need to do in Shapley is that you need to find if I add A to any coalition of a smaller size, let's say add A to B, add A to C, or add A to any combination of other pairs. So what would be the marginal contribution of A? And computation of Shapley values and Shapley interactions is again an exponentially hard problem. So you need to evaluate this function, the neural network, exactly q to the power n many times. In this case, 2, because we are talking about binary. So my student Darren Sui found out that there is a really neat connection between the Fourier transform of the model, which we were able to find very efficiently, and the Shapley interactions and Shapley values through something called a set-Mobius transform. Set-Mobius transform allows us to start from Fourier, which is the global understanding of the AI model, to zoom in into Shapley, which is a local understanding of the AI model around a particular sequence. And what he showed is that you can actually do one level of sketching. You can sketch your AI model just once, represented in terms of the top K Fourier transform of the model, using a computational complexity that grows, again, linearly with the sequence size, with the number of mutation. And then once you do that, you're done. So you're going to be able to find the Shapley values for any future sequence essentially free. The computational complexity doesn't grow with n. It doesn't have any dependency with the size of the sequence. So one trick that we used was this idea of amortization, therefore. So sketching, paying one cost to be able to sketch the model, and then you're going to use a sketch to be able to explain future sequences much more efficiently. We have made the software publicly available. We call it SHAP0 because you need to pay zero cost for any future sequence. So if you have any problem where you are interested in understanding if you have a genomic problem, if you have a proteomic problem, doesn't matter, where the input is a sequence, output is a property of that sequence, happens in many areas in biology, then you will be able to use SHAP to be able to explain this black box in terms of input-output relationship much more efficiently than existing methods. So we tried it on transception, which is an autoregressive model, like next token prediction in biology. It's around 700 million parameters. It was already one of the biggest when we started this project. Now it's too small. Regardless, what we saw, so the question was that, can we see how transception learns the interactions between these mutations in GFP? What we saw is that you can basically input many sequences. In this case, we inputted 200 sequences in the training data. And we saw that the SHAP values that we find very much matches with the ground truth kernel SHAP. It's not the ground truth really, but the baseline algorithm here. And in particular, the correlation between these two was around 0.97. But in particular, we can get to the same explanation 3 times faster. But more importantly, what you see is that the computational cost of SHAP in this case is essentially almost flat. So if you explain the model, so if you increase the number of input sequences to even larger, so the computational complexity in this log plot is not going to change at all, but kernel SHAP is going to have a linear computational complexity for sure. So this gap is going to only increase. Things are even better when you're explaining higher order interactions. So for Shapley interaction, we are seeing something around 7 to 10x acceleration in explaining the AI model. Now, everything that I said so far was on the black box, like explaining black box models. Sometimes you don't have access to AI model. Let's say AlphaFold 3, you want to use something like this. Sometimes you do have access to the internal nodes of the model. They're open access. So for example, in the case of the protein language model. And the question was, can we use the internal nodes now instead of taking a completely black box approach to be able to understand the computational circuitry inside a protein language model? Very similar to GPT and large language models, the reason that these models are really hard to understand is that the nodes, the neurons inside these models are polysemantic. Each neuron can have different meanings, can do different functions at the same time, and this makes understanding large language models very hard. So we were pretty much inspired by some of the recent work in Anthropic and developed something called a replacement model, which is essentially another AI model, which is much larger than the original model, with the big difference that we use 2 tricks. One is lifting the dimension of latent space to a higher dimension and impose the sparsity. So we hope that by doing so, if there's a polysemantic neuron, this is going to be expanded towards many monosemantic neurons, so that if you look and zoom on one of the latents, we will be able to understand what is the biological meaning of each node. And in fact, this is what happens. What I'm showing you is actually a real case of a protein language real mechanistic tool that we have developed. This particular node corresponds to a HRD motif, which turns out to be related to some catalytic activity of the kinase proteins. And what we do is that we basically search over each neuron, see what sequences are fired, and then come up with some sort of an explanation about each term. What made this very successful was that we used some particular method, which is called cross-layer transcoders. So these are transcoders, or something like autoencoders in the good old days when we were doing dictionary learning. But the difference is that now we apply it at every layer and cross layers. And being cross layer is really important because these computational circuits across, they really vary, they really are interconnected across the layers. When we compare the performance to a version which is called PLT, pair layer transformer, when you kill the connectivity across the layer, you're going to see that the prediction performance of this explainability method is going to drop. So we named it ProtoMech because it's really mechanistically explain what is going on in a protein language model. We use them to be able to engineer new and better proteins. Here we see that you can actually take a circuit inside the protein language model, crank up the weights that corresponds to the fitness value, and we see that we can go from the wild type protein to design proteins that have a higher fitness value, again, compared to PLT and compared to directly operating on the node level instead of this latent space that we have defined. More strikingly, we can actually explain what happens in a protein when you do a mutation. Here, this is a circuit from ProtoMech. GB1 is a FC binding protein, very is a standard benchmark in protein engineering that we have used. ProtoMech thinks that the reason that GB1 is stable is because of the tryptophan at site 43. It makes it stable. We were really curious to know why there is a mutation at site 24 from alanine, again to tryptophan, A to W, that makes this other variant very kind of have a high fitness. And we immediately realized that ProtoMech predicts that there is going to be a new node born because of this mutation in the computational circuitry of protein language model, ESM. And it explained that what happens as a result of this mutation is that there's going to be an inward-facing residue, which makes the entire GB1 protein more stable. And that's the reason that AI is thinking this mutation is going to make the protein higher fitness because GB1 becomes more stable. That was really striking to us. And this is one of many, many examples that we have explored in the paper, which are really a step towards better understanding at least a fraction of what AI is doing in these protein language models. So we have made the software open source. We have so far trained it on ESM is a model by Facebook. Now there are many other variants that are coming out. We have trained it on 8 million parameter model, 35 million parameter model, and we are starting to understand that the scaling to full scale of 15 billion is a challenge because the computational complexity of learning these transcoders scales quadratically with the number of layers. This one is 12 layers, this one is 20 layers, this one has much more number of layers. So we are developing some approximate algorithms to be able to now handle the quadratic cost of these mechanistic tools that really need to reason across the layers. Okay, so now I just wanted to, after this, I just want to highlight the last piece of stuff that we are doing. Okay, so another direction other than interpretability and mechanistic interpretation that we are very excited about is the question of fast protein generation. So there are, just like GPT, there are lots of autoregressive models in protein where you can generate proteins. The question that we asked was that can you generate proteins faster? Because for high throughput experiments, you might need to create a really large assay and screen for a large number of sequences. How can you make the generation process of proteins faster in autoregressive models? We knew that Google has an algorithm called a speculative decoding algorithm, which is kind of an interesting statistical trick to be able to generate proteins faster. The way it works is that, let's say you want to generate a sentence like LLM. Instead of generating using a target model, you're going to use a smaller version of the LLM. I call it a draft model, which is smaller. For that reason, is going to predict the next token much faster, generate the next token much faster. What the speculative decoding does is that you generate using draft model. It's pretty cheap. And then once you generated that, you're going to validate using the target model. Now, since you are validating it block by block, validation is going to become faster as much as the block size is. In this case, if I'm validating this tree guy, so I can do one pass through the LLM and then try to validate that if this particular three-added tokens is good or bad. So we thought, okay, let's just bring it into protein. Nobody has done it. So we trained a speculative decoding algorithm on an autoregressive model for proteins. We were quite happy about the acceptance rate, but we realized that something interesting is going on. So this is the distribution of the proteins that we expected to happen. X-axis is the likelihood. But then when Tom, my student, was looking at the likelihood of the version of the speculative decoding algorithm, he realized that there's a shift. It seems that there's a drift going on towards the peak of the distribution. In other words, we are generating sequence from the original target distribution, but they are more narrow. So we are missing some of the high quality proteins. It was a curious case. We didn't know how to explain it. We didn't name it. We just went ahead and come up with a computational biology trick that helps us to solve this problem. But just a few weeks ago, there was a paper which was actually gave a name to this in the context of LLM. They called it attention drift, which resonated with us a lot because it seems to be what is happening is that as the draft model is generating next sequence one by one, when the protein or when the sentence becomes too large, the draft model becomes too much biased on the tokens that it has already created. So for that reason, you can see that it generates sequences around the mode and less sequences around the tail. And these ones, it turns out for the protein case, the ones that we are actually very much interested in. So what was the solution? So Tom was like, okay, can we use some a priori knowledge about the protein to be able to improve the generation quality of that? So where is this a priori knowledge is coming from? Well, multiple sequence alignment data. So what we can do, and what we did, was that we can take the sequence that we are interested in generating, like maybe a new GFP. We can look at all the protein sequences in this family. We just talked about how we can do multiple sequence alignment fast. We just need to do it once. And then we said, okay, let's draft multiple sequences now, and then do a k-mer filtering step, which is going to accept or reject the sequences drafted by the draft model based on the k-mer statistics of the MSA. Okay, so if the drafted sequence match up with the k-mer statistics, we are going to accept it. If not, we are going to reject it. And what we saw is that in this way, we increase the quality of the sequences generated by the draft model, and the overhead by this k-mer filtering would be really small because you can do all of these processing offline. And as a result, what we see is that in our new algorithm, which we call it SpecMer because it's a speculative decoding, like k-mer type idea, is that the SpecMer distribution kind of shifted to the right, and now we were able to see these high quality kind of proteins that are generated. So we tried this on ProGen, which is kind of a well-known autoregressive model for protein generation. We used the 151 million one for drafting, the one with 764 as target. And what we see is that when you do vanilla speculative decoding, the acceptance ratio is okay. So the target model is accepting around 80 to 90% of the drafted sequences, but the likelihoods are not good. So the high quality protein sequences were missing. So when we did SpecMer, sees the number of drafted sequences and like the batch size of 3 and 5. What we see is that not only the acceptance ratio is going to improve, but you're going to see that the likelihood, the average of the likelihood for the protein sequence is also improved. This suggests that the quality of the protein that is being generated also improved. So we get kind of two advantages at the same time: better quality proteins and also around 25 to 30% speed up by knowing something about the biology or the prior information about the protein. Okay, good. So I think I have 2 minutes. So let me just wrap up. So I mostly talk about 3 things: how we can take a black box AI model and try to explain it more efficiently when you're doing query to input output. Second thing, which was the SHAP0 algorithm. Second one was mechanistic interpretability. You can actually open up a protein language model and understand the computational circuitry inside them. And third one was that you can do fast generation if you know something about the statistics of the protein. These are most of the papers that I presented, but we do lots of other stuff in the group. Something that I want to highlight here, because it's mostly around data acquisition. So we use the sparsity and low dimensionalness of cryo-EM images to be able to develop better acquisition systems. We also work on some agentic system, which I didn't have time to talk about. But these are all, it's a very fast-paced field like AI and bio, and I think it is still very relevant to think about bringing algorithmic ideas, hashing, what we know in signal processing and coding theory to be able to advance the field, not just relying on AI models for prediction, but actually open the box and understand them from a fundamental perspective. So yeah, I just want to thank the group and all the funders and open up for questions. Thank you. Yeah.
Thank you so much.
Thank you for the talk. I'm not a machine learning expert, but when you explained proteomics, you make a consideration that there were issues in scaling the number of parameters. Right. So my question from a non-expert of machine learning field is it really needed to scale and the number of parameters? And if so, which advantage do you expect, for instance, in your kind of specific application?
Yeah, we definitely need to scale. So there is a really nice chart. If you go to the GitHub of ESM, there is a fundamental, like in terms of prediction ability. So ESM to 35 million to 150 million, there's a huge leap here. Things won't change a lot here. And then from 3 billion to 15 billion, there is another really jump in terms of prediction accuracy. Now, this is the original model. It's a good question to ask. If we apply this mechanistic tool on these models, are we going to gain more and more? We have seen that going from 8 million to 35 million has been really, it's a game changer. So I don't know about the rest. Hopefully we'll have a good answer to you soon. Yeah.
Thank you.
Yeah. Thanks.
Any other questions?
Oops. Thanks for the talk. I know you're incorporating multiple sequence alignment in your analysis. I'm wondering if anyone in the field has tried out incorporating phylogenetics, like how these sequences are related to each other, and use that.
Yeah, it's a good idea. I'm not aware of anybody, but I think it's an interesting idea. Currently, I mean, it's a good observation, right? This is very much protein dependent. Like phylogeny can also expand it to a wider set of protein sequences. Yeah, that's an interesting idea. Nobody has done it. Yeah, as far as I know. Yeah.
Anything else from the audience?
Great.
Okay, thank you so much once more. So I think we can continue with our next talk. Okay.
Oh.
Have you shared the slides? Are you using your laptop? Okay. Sure.
Thank you.
Thank you so much. I just have to do this again.
Sorry.
No worries. And now I've done it. Okay, we're good.
Okay. Thank you.
So I'm gonna check something here. So if you could only wear this.
Yes. Okay.
Okay, so in the meantime, let me introduce the speaker. So Davide Conficoni is currently an assistant professor with the Dipartimento di Elettronica e Informazione e Bioingegneria Politecnico di Milano. His research interests focus on the design of fundamentally more efficient and scalable computing systems by domain specialization. This work is pursued by hardware-software co-design, jointly reshaping algorithms, compiler infrastructures, architectural abstractions, macroarchitectures, and heterogeneous systems.
Thank you for the introduction. Hi, I'm going to bring you a slightly different perspective, but yeah, let me just skip this. I'm kind of trying to work at the interdisciplinary area, and being a computer system expert, I'm also diving to another area, which is whiskey aging production. We're going to run a workshop if you're interested in TUM, 2nd and 3rd of July. And interdisciplinarity is fundamental, and I think that this workshop is kind of doing great in bringing a very different area of expertise. especially if you look at an observation that was made by another researcher, Sarah Hooker, who was introducing the concept of hardware lottery, where basically the observation was that the field was advancing because the idea that were winning were just because they were mostly compatible with the available hardware, like GPUs. While from her perspective, this is kind of fundamentally wrong because it's kind of, they are not superior in terms of research perspective. And from her perspective was machine learning was an example. And she's arguing from the fact that we must think of a new way of doing and exploring different hardware software algorithm combination, or basically what I called as a co-design. And co-design is fundamental because you must have a different field of expertise. And in a keynote last year, HPCA, Cliff Young advocate for this co-designing capability. Especially he was co-designing Anton, one of domain-specific system for molecular dynamics, where he was basically saying that it's fundamentally important to interact with domain experts to build system that can be bring 1000x improvements. And in doing so, I want to talk to you about a specific domain, which is the regular expression matching, which is slightly different because it has both application in security, but also in motif search into the bioinformatic fields. However, regular expression matching is something that is embarrassingly sequential because you can proceed with your computation only if you know the outcome of the previous operation, which is a problem. And generally in the literature, we see a solution that struggle to create solution that balance performance, flexibility, because probably we are discovering new motifs that we want to search in our data. And we want to be flexible into the motifs that you are looking for. And of course, energy efficiency or what Onur was showing, miniaturization of devices. And this brings me to and us to this this fundamental question, which is if we could find a unified solution that could bring a sort of balance in this trade-off. Because if you analyze more in depth from the computer architecture perspective, what you see is that CPU, which are the most flexible engine ever, there are in-memory solution, and there are also kind of solution that for our FPGA community are widely spread. So take the logic of your computation and embed it into the reconfigurable fabric. But this is kind of inflexible. So what we are thinking is to try to adopt a different approach, try to build something that exploit hardware specialization, but as well exploit adaptability, so software programmability. And in doing so, we create a domain-specific framework, which is built out of a language to express operation, an instruction set architecture, a compiler to basically translate automatically, and to a microarchitecture. And the fundamental idea is that if you look at regex patterns, you can see them as a language. And a language has an algebra, which has certain properties and certain operation, which then must be nailed down to the very specific operation of your algebra of your language. And then they can be translated into a sort of binary representation. This is good because then you're empowered to create a language. But also, you have to look at the way that this language is executed. There are two fundamental approaches. One is a speculation, and one is enumeration. So we started building something into the speculation-based approach with this latest generation, which is Alveare. And the fundamental thing in Alveare was that we build everything custom. So, and in customizing everything, we started even from the program memory and to the data memory. And the program memory basically was designed to be trying to prefetch and foresee all the possible execution flow, all in parallel, because then what you want to try to achieve is to extract this multiple control flow and parallelize them through vectorial execution. All of this orchestrated by a control unit that basically manage the issue of speculating, so going that first and then get back if the result was not correct, which is great because this builds a fundamental unit of a microarchitecture that we can then try to scale out to have parallelization in terms of regular expressions, so patterns that we can search and data that we can analyze. So a sort of divide and impera approach. And this was great. So we started analyzing a different perspective. Of course, being a computer architect, energy efficiency and speed were important. And in trying to analyze with different benchmarks that spans from network intrusion to proteomics, we were able to see that scaling out the core number was kind of more efficient. But most importantly, compare against other solutions that were based on GPU, or better, a silicon-proven accelerator for regex matching that was featured into the Bluefield 2 DPU, we were able to achieve energy efficiency dramatically better, and as well in terms of time, of course. So this was one way, one possible way. We explored in parallel the enumeration with another framework called Cicero. Cicero here has a slight different language. So the same language of expressing patterns, regex, but based on an early paper from Ken Thompson and Russ Cox, where basically the operation needed to represent your language are much fewer, because the idea is that you want to explore in parallel, enumerate, all the possible alternatives. And this was the minimal set of language. And out of this, we started building our architecture organization with the basic unit, which was an engine that then featured a core to perform basically the comparison and a set of FIFOs. These FIFOs basically are our possible alternatives. All of this process, matching process, then is orchestrated to be executed in a lockstep fashion by a centralized controller. This can also be scaled out in trying to extract more parallelization from all this possible control flow through a multi-engine. Interesting, we also compare against terms of speed and time, and we were able to achieve remarkable improvement against CPUs, both embedded and mobile ones. But there was a problem when we study how we created basically the architecture organization. This architecture was not scaling. There was an issue in adding more cores was basically worthless. And in trying to do so, we started from the bottom now. So in co-designing the solution, we started looking at the architecture, and instead of having multiple FIFOs per a single core, we basically redesigned the architecture to have one FIFO per core in trying to scale in a better way. Also, we were working at the full system design stack. And Cicero was featuring its own compiler, which was a sort of monolithic. That's why we redesigned this compiler infrastructure, leveraging MLIR, so multi-level intermediate representation, and try to focus more in decoupling the way that we were approaching the problem of compilation. One tailored just for the high-level language, so the regular expression language, and then adding another dialect that was more focused onto the low-level backend that was Cicero microarchitecture. This was great because we were able to extract more optimization, and because we were able to avoid an issue that was called parameter lowering. So our monolithic structure was not able basically to introduce very basic optimization that thanks to this decoupling, we could introduce, basically understand better the program and the hardware itself. And this was great because we were able to improve over the previous work. These were the domain-specific framework that we were creating in trying to optimize the process of regex matching. But they were extremely tailored for a single regular expression. As you might know, if you analyze a stream of data, then you need multiple patterns, not just a single one. So there was a bit of limitation of the way that we were building this architecture. That's why we started looking at this multiple regex. And we made an observation that there were multiple shared patterns across different positions of our rule set. This was great because in that way we could probably extract these common sub-patterns across the different rules and try to use it as our advantage. However, if you do this, you have to pay attention because this common sub-pattern must have a very predefined specified path, because we don't want to introduce patterns that were not there before. So that's why we introduce a new concept of activation function in our rule set representation. In that way, we were able to distinguish to which basic set of regex this automata were belonging to. We started so redefining a new formal way of looking at automata, where we basically have something that are states, an alphabet over which we are performing our recognition, the states that are initial, and the states that are final. All of this with a state transition function. So far, if you have a slight familiarity with automata, it's a basic automata. But then if you merge multiple automata together, what we introduced was a concept of the fact that these automata were belonging to different original rules. And in doing so, we must also keep track to which automata originally they were belonging, which was our contribution in basically create this new formal model for multiple regex representation called MFSA, which basically was looking at the data representation, was able to introduce a gain in terms of compression. However, if you take a look at the different benchmarks, Pro, which is Protomata, a proteomics benchmark, was one of the most complex because our technique was able to have some compression gains, but not as for the other application domain. So we want not to have just theoretical benefits again, because we want to have this regex and we want to automatically generate automata out of our regex that then must be merged automatically. We didn't know which were a priori the optimal merging capability. That's why we created a way to automatically merge a different quantity of patterns in our original rule set through a compilation framework. And then basically what we did was also to add an execution engine, because the way that you represent your automata now is fundamentally different. And this difference introduced an issue. That's why we also add to this new execution engine, based on the state of the art, the capability of recognizing to which automata originally this was belonging to. Starting from the basically baseline state-of-the-art approach, we were exploring the different parameter because this was an experimental parameter, which we didn't know a priori what was for. We were collecting a bunch of data, but the most important thing were twofold. One, of course, we were able to achieve better performance in terms of raw execution time. But the most important thing is that with this way of representing motifs, you are able to save computational resources. So you need less computational resources to basically perform the same task in a better way. And that was great because we have also a gain in speed up. So this was the fundamental research question, which is not basically a unified view because there were different projects, different things. But the way is that we are starting to build out of this different direction, one thing, which is unifying now the compilation and on how we can represent basically regex matching. And basically you have an heterogeneous set of backend. We have CPU, GPU, FPGAs, and many others. And then we have our own software, which is the basis for us because our software then is in charge usually to, given an automata representation, compile for our target backend architecture. And right now we can find several different compilers across several different backends, which is okay, but you might lose few possibilities of having a unified approach and that all the others try to work into the same direction. That's why we can use and leverage what is called MLIR. And we are starting working on this MLIR, basically compilation infrastructure that, given a regex dataset, is started working and building on top of our previous algebra dialect for compilation, novel dialects that we are building for automatas, and then merging multiple automatas to then have a standard, either standard compilation infrastructure or custom for different backend. But try to unifying the first path of this pattern matching approach, which can, in a certain way, help us in standardizing that way that we are moving. And this was one possible direction, but this MFSA also had another advantage in terms of execution time. So this means that at the hardware level, there are possibilities to explore parallelization. And for instance, you can find a general purpose processor, which of course, our MFSA works great in terms of compression and execution. But then we have a domain-specific solution like Darwin, like we have seen also in memory solution and different others. But they are, generally speaking, they are focused on automata representation that are more traditional, which are, however, incompatible with an MFSA representation. That's why there is this other question that we are working on is if we could find a way to map this new fundamental way of executing regular expression matching on domain-specific hardware. And we are creating an automating toolchain called Sparks that basically generate automatically the RTL based on a novel primitive at the hardware level to represent this computation, also to be plug and play in any streaming XI accelerator. Stay tuned because we are trying to work on this and basically create a paper out of this. With this said, I'm kind of going towards the conclusion. This is a huge group effort from many different people that have been collaborated on this research project and research direction. And to wrap up, just to make a turnaround of what I presented. We have regular expression, which was something that is embarrassingly sequential. And we have to find a solution that try to unify this different trade-off in performance and flexibility. I presented you how we were tackling this by domain specialization at the computer system stack, but for a single regex, and how we are trying to tackling this at the algorithmic level to represent a novel way of rule sets. And this is basically the next step, the first step to create more novel and significant better systems in terms of regular expression matching. Said all, thank you for your attention. I don't know if there are any questions in here.
Very much for the presentation. Do you have any questions?
Hi, thanks for the work. It's great to see that you went really through the full hardware design and then MLIR stack. And also, I think it's a very good approach and good example of using MLIR here. When you talk about this domain-specific reconfigurable architecture, I see where you're trying to go, but do you think the way is to go towards basically building tons of different ASICs, or do you have a bit of can you explain a bit more the philosophy behind? Because I'm interested.
Yeah. As of now, there was not a concrete win, in my opinion, to have one ASIC or not. And also, if you look at for instance, the proteomics domain or the bioinformatics domain in general is kind of evolving a lot. So the idea was having FPGAs, of course, are a prototyping platform, but then you can even experiment. All of these architecture are customizable. Not every workload needs the same parallelization. Not every application needs wasting resources, wasting power. So as of now, FPGA were the ideal platform for experimenting this direction and also to be adaptable. One idea was if you take the blue field is a sock, and this sock is extremely fixed, the regex acceleration in silicon, which is good, but then they lose few opportunities. So from my perspective, there are two parallel directions. One direction that I'm wishing to explore, of course, is to go towards creating a silicon-based architecture. And for instance, trying to use open source tool chains. This is something that I'm thinking and I'm trying to work on it. I don't think is the final solution. So it is a solution, but as of now, it is not mature enough, in my opinion, to say, okay, we are done and let's go just for silicon.
That makes sense. Thank you.
Thank you for the question.
We have another question. Okay, then I think we can go on.
Thank you.
Thank you so much once more. So let's go to the next one. It's John. You're already. Okay. Okay. So John, you can wear this.
Sure.
And I can briefly introduce also John. So John Furtina is an assistant professor in the Department of Computer Science at the University of Maryland, where he leads the STORM research group. He received his PhD from ETH Zurich very recently, 2025, where he was advised by Professor Onur Mutlu, as it was mentioned before. His research spans a broad range of problems that address the computational challenges in biological data analysis. And I think John can now proceed to let us know more about it.
Sure. I think I'll place it here because I want to see the screen. Okay. Thanks for the nice introduction, Konstantina. And I guess probably you already seen some of my slides earlier today by Onur Mutlu, but I tried to remove and add some new slides, so it will be hopefully much newer content for you. So with that, let me get started. I'm John, as Konstantina mentioned. And today I'll talk a lot about genomics, basically the genomics part of the bioinformatics research. And we basically want to understand and analyze genomes because they are useful for many applications. We can reveal key insights by analyzing our genomes. For example, we can design and prescribe drugs by understanding how you'll react to them, or even change the content of your genome, maybe to disable some functionalities or enable some of the genes that you don't have through genome editing, or maybe detect pathogens growing among us, and by doing so enable rapid surveillance of disease outbreaks. And to enable all of these applications, we want to look at our genomic picture, right, and analyze it. And what I mean by genomic picture is this huge string of these DNA letters. In the case of human genomes, we have about 6 billion characters in a single cell, and we have trillions of cells. And of course, this data is not in a human readable form initially. It's a bunch of biological molecules sitting in our cells. If you want to generate data out of it that in a format that we can analyze, we use these sequencing devices that take these biological molecules as input and generate some sort of raw genomic data. So this raw genomic data, for example, can be in electrical signals, and this can be translated using AI models to ATGCs. But unfortunately, we don't get this data in a full single format of our entire genome or entire chromosome. Rather, we have very small pieces that we call reads. So these reads are extremely small compared to our entire genome length. These are about from a few hundred bases to a few thousand bases. And to make things worse, we don't know where these reads are coming from. So this is really like a giant jigsaw puzzle that we're trying to solve. And we may have a reference picture or reference genome that we can look at that is previously constructed so that we can identify the origin of each read. and also the exact differences between a read and the reference genome. This is because we may have mutations or the errors in the data. And because of course, like all of these issues, we redundantly generate large volume of data, maybe from terabytes to petabytes, so that we can reconstruct our own genomic picture from scratch very accurately. So in a sense that we're really dealing with a massive and noisy data sets from terabytes to petabytes. So then basically deconstructing this, the genomic picture, identifying the origin and the exact matches is usually known as the read mapping step, as Onur mentioned earlier. And it has many downstream applications. For example, we can use these origins or the exact differences in the variant calling step to identify what sort of mutations you have so that you can perhaps link them to certain diseases or in metagenomics. But when you look at this pipeline, we're generating this large volume of raw genomic data, right? It's electrical signals, perhaps, and maybe you can consider that the whole data set is initially in the size of terabytes. And as you basically move towards the next steps, what happens is that some portion of the data becomes useless. This is mainly because maybe we have a low quality data, or maybe we're applying certain heuristics that they don't get used and so on in the later steps. And actually, if you really have a messy sample, or maybe in the case of metagenomics, if you're really interested in a certain sequence, a significantly large portion of the data that we're analyzing can end up being useless for our analysis. But the issue is that you're moving this user's data across many steps. You're computing this user's data, which means that you're actually wasting money generating this data, wasting time and energy computing and moving this data. And these steps are not necessarily cheap. We're spending hours to do this, to do a single step in the analysis, and these numbers are only for a single person. So they get extremely worse if you want to do a population scale analysis. To make things even worse, and maybe a bit relatable to reconfigurable computing, this pipeline that we're seeing in genome analysis is not really balanced. So depending on the data type that you're generating, depending on the technology that you're using, depending on the analysis that you're doing, the throughput will change, right? So what sort of algorithms do you need to use, right? Or what sort of computation do you need to do? So all of these issues then put a limitation in the application scope and the accuracy of our analysis, especially when you need energy efficient analysis for resource constrained devices, for example, drones running in agricultural fields, or urgent analysis that you need to do within minutes, or maybe large scale analysis without sacrificing accuracy and performance. So in our group, in our STORM research group, we basically tackle all these issues so that we can enable a fundamental better integration of biological data analysis in our everyday lives, because it has many implications. For example, we could do more frequent food profiling to make basically our food much safer, or continuous analysis of our health, and even urgent analysis for neonatal screening. But in today's talk, I'll quickly focus on only one direction that we're focusing on, which is really enabling the real-time and portable analysis by analyzing the raw genomic data. So to show you what sort of sequencer that we're using, although it was mentioned earlier, so we're using these devices that are called nanopore sequencers. And these are really small devices, right? So this is a handheld device that you can carry with you, so that you can sequence DNA. And this is, at a very high level, this is what happens, right? So you have this DNA molecule, you have a 3-layer solution, top layer is negatively charged, the bottom layer is positively charged. And DNA, due to the electric field, it basically moves from the top side to the down side. And as it does, it disrupts the ionic current flow that we have inside this tiny pore that is called nanopore. And as we do so, we basically report these ionic current disruptions as electrical signals, which is generated as a certain throughput, so that we can do some sort of analysis. And if your analysis is fast enough, you can actually analyze this data as it is generated, which we call real-time analysis. And one big benefit is that as you do this real-time analysis, you may realize that the molecule that you're analyzing at a time is maybe useless for you. So then you can tell the sequencer that you don't want to keep sequencing that molecule, so that you can save money from sequencing that molecule, but also time, not sequencing it and also not analyzing it and so on. And actually, we're seeing an interesting trend nowadays towards using portable sequencing. And I'll give you two interesting examples. So this was actually quite popular 2 years ago. A high school student, unfortunately, his pet fish died. And after basically that sad event happened, actually, he wanted to raise money because he wanted to analyze the genome of his pet fish. And basically, he raised the money to buy a sequencing device, but also to do the analysis. And to make things even more interesting, this was the very first time that this particular fish, which was an angelfish, was sequenced and also analyzed so that its assembly was generated for the first time. So it basically initiated a spark in the community, and everyone was really interested in this development because a high school student could do it by itself, by himself, maybe collaborating with some the biotech company that was nearby to him. But also with the emergence of AI, what's happening is now, like this is a very recent example, as I think this tweet is from a month ago. Another person bought these portable sequencers and asked Claude, saying that, oh, I have this nanopore, so how can I sequence my own genome? And then basically he followed the instructions and then he sequenced his own genome, analyzed it, and generated some data. But later, of course, he realized that the data quality that he generated was terrible. But I think what this is showing you is that I think there is now a paradigm shift that's happening, right? So it's not like I think the industry is going to be generating data, and then it's not going to be up to the industry to analyze this data, right? The individuals are going to be generating their own biological data, or maybe the data in the out there, as if maybe they are recording videos and uploading to YouTube. I think this requires us to rethink how we should redesign these databases, the public databases, and also how we should be enabling portable analysis in a portable, scalable, secure, and private way. So with that, I'll quickly show you some examples of what we did towards enabling this direction. Onur already mentioned our raw hash work, and we also published several other works in this direction to analyze these electrical signals without translating them, again, to enable new applications and to make the analysis much more accurate. So remember this pipeline that we need to translate these electrical signals into human readable text, DNA text, so that we can do the further analysis, right? So we asked the question, actually, do we really need the translation, right? Maybe it is to do some of the analysis. And the question that we ask, actually, can we directly analyze these electrical signals without doing any translation? And for this, of course, we need to deal with the noise in the data. And we have noise. Maybe one type of noise that we have in the data is when you want to sequence this particular molecule, what's going to happen is that you're going to generate some series of electrical signals that represent the content of this molecule. But what happens is when you want to sequence exactly the same molecule again, you'll generate some signals, but these signals are not going to be exactly the same to what you generated earlier. But the observation is that we're going to be generating similar raw signals when we're sequencing the identical molecules. And if you want to identify the similarity, because it really at the core of bioinformatics is perhaps identifying similarity between maybe target and query sequences so that you can do further analysis. One idea is really to do a vector similarity search by nearest neighbor search. But this becomes too costly for large genomes. And as this figure was shown earlier, this figure shows the computation speed relative to the data generation speed, right? And if basically this means that if you're below 1, then this means your computation speed is slower than the data generation speed, which means you cannot really do real-time analysis as the data is generated. So we wanted to solve this issue, and what we did, instead of doing costly vector search, we said, oh, let's do the hash-based search that like everyone is doing in the bioinformatics space, but what they are doing is they are doing it on the sequence space, right after base calling. So of course the challenge was if you want to do fast search, we need to generate perhaps exactly the same hash value so that you can do quick exact matches. But how do you do so when the signals are different than each other? For this, as Onur mentioned, what we did is we actually quantize the signal so that we eliminate slight differences between them. And the quantization is really means you basically assign the close-by values to the same bucket so that you eliminate those differences. But the challenge is, of course, the signal is not really uniformly distributed across its entire range. So what we did actually, we learned from the nature, the type of the noise that we have in the signal data and integrated this to the bucket generation idea to make the bucketization, the quantization, the adaptive. And this is really the adaptive nature of quantization. And this is adaptive in a way that in the future, if the sequencing technology changes, you can go ahead and analyze the signal and change your quantization strategy again, depending on what sort of distribution you have. Well, of course, we don't really want to use a single quantized value for matching, because then it would cause millions of matches in the database. What we want to do, we wanted to make the hash value a bit more unique so that we have fewer matches in the reference genome, but still we want to make them accurate. And for this, what you can do, you can increase the range that these quantized values are spanning by packing them together, right? So if you pack them together in a single value by still keeping their order. So this means that keeping order gives you an accurate match, and packing them gives you fewer matches. So you can use this packed value as a single hash value so that you enable fast and accurate match. And the key contribution of raw hash is, this is really the first hash-based search and indexing mechanism for raw signals, so that we can bring all these optimizations, all these techniques that people develop over the last 40 years in the signal space. And this enables us to achieve real-time analysis for the first time for human genomes. But also, by doing so, we're still more accurate than the previous nearest neighbor search mechanism in all data sets. Although this is perhaps not so intuitive, because hash-based search is an approximate mechanism compared to maybe a nearest neighbor search. This happens because when you're doing nearest neighbor search, you still need to rely on certain heuristics, so that this nearest neighbor search becomes still practical in such a massive space, like search space in bioinformatics. So these heuristics actually are preventing this the nearest neighbor search approach also to achieve accurate analysis. And since we don't have these costs when we're doing searching, we can disable those heuristics and still achieve accurate analysis. So raw hash is fully open source, and it also has a new home in our new research group. So we keep developing raw hash, keep integrating new functionalities. Recently, we provided this API that enables us to talk to these nanopore sequencers directly. So you can plug the nanopore sequencer to your computer and let the raw hash run as the sequencer generates data. So before perhaps I start concluding my talk, I want to give you an idea about basically what this type of direction enables. So you may be saying that, right? So, okay, so we enabled the real-time analysis for the first time for human by directly analyzing raw signals. So what, right? So, okay, what are maybe like the implications of it? And one implication is that by making certain analysis practical and accurate, you can also enable new applications that didn't dare in the raw signal space. And this is a very good example. Raw sample was published 2 months ago in the Bioinformatics Journal. And what we do here is that, as we do in raw hash, so we can map these signals to our reference genome, right, using this hash-based seeding and mapping approach. But then you don't really always have reference genomes available, or maybe you don't know what you're looking at, right? So in this case, what you can actually do, you can find the overlapping regions between reads, and then you can basically stitch them together to create a consensus, or we call assembly, to create the reference genome maybe from scratch. But of course, we could not do this without doing base calling. So we want to do this really on the raw signal space. So I'm not going to really describe you details of our mechanism, but what we really did is we built on our prior works, raw hash and raw hash 2, added some optimization, some filtering techniques to reduce the noise in the data so that we can find many useful overlapping pairs. But what I want to show is that this figure shows the runtime of 3 different configurations. And all of these configurations are doing the same work, basically same task, finding the overlaps. But the real difference is that the raw sample, our work, does this task without doing any translation, any base calling. And the other 2 first do the translation, then the overlapping task using CPUs or GPUs. And the biggest, I think, the observation is if you basically eliminate the translation, the base calling from the analysis, you can achieve a significant speed up to 60 times, right, by doing to do the same task. And our implementation is basically CPU-based, and it's still 2 times faster than the GPU-based base calling. So basically, what would happen, right, if you had a GPU implementation of raw hash, and what sort of benefits you would have maybe in different hardware platforms. So one last thing that I want to show is that we also realize that the raw signals are extremely noisy, and they may need also noise reduction or error correction. And so we also wanted to propose some mechanisms to correct errors. So when you're analyzing raw signals, you perform a set of steps. For example, segmentation. You identify the portion of the signals that are generated by sequencing a particular molecule at a particular time. And this segmentation, then what it does, it basically converts these raw signals into a series of values. And you can also then do quantization to generate the hash value and then perform the query in the hash table to find the matches. But what happens is that if you have an error, maybe due to the noise in the data, or maybe due to the mistake that the segmentation algorithm makes, some numbers may change because of that error, which will lead to completely different hash value, and which may give you a maybe false hash match. So what we realize is that the error, the noise in the data also reduces the accuracy, the propagates to the later steps in the analysis. So what we did is we wanted to understand this error, or understand what an ideal sequence of signals look like, and also understand the error type, and learn all of these in a hidden Markov model so that we can understand whether the real signal that we're analyzing at a time contains an error or not. So to do so, we generated all possible sequences of a certain length, and then generated their ideal sequences. And by doing so, we integrated this in a hidden Markov model, and this gives us some sparse HMM, which gives us basically the ideal look of the series of raw signals. But we also wanted to learn the errors that we can have in the signal. To do so, we use real signals, and then do the segmentation and apply in a downstream application such as raw hash, and then looked at basically the accuracy of raw hash after basically doing the correction with our sparse HMM to integrate some probabilities that reflect also the errors in this HMM. So the final resulting HMM contains the probabilities both for both the error-free series of events and also for the erroneous events. And we use this HMM to correct the signals before performing further analysis in the application. So we integrated our tool, and we stopped calling our tool CERN. So we switched to Crane based on some feedback that we received. So that's why you'll see the Crane instead of CERN in these figures. So what we realize is that the error correction consistently provides better accuracy in the analysis. And this is especially true for one particular configuration. So this figure shows the accuracy in different data sets. So like each of them are E. coli, fruit fly, human, the larger data set as we go towards the further side. And all of these use the latest chemistry in the nanopore. And here on the x-axis, we use different segmentation algorithms or segmentation configurations. Campolina is, for example, is one of the most recent ML-based segmentation algorithm, and Scrappy is an earlier one. So the issue with Scrappy is that as a new chemistry comes in, you need to basically change the algorithm a bit and learn again or understand the related parameters so that you can integrate it to Scrappy. So there's some manual work. And the issue with it has been always, there is some Scrappy algorithm that's developed for prior chemistry of nanopore, but it didn't really work, as you can see in terms of accuracy for the newer chemistry, right? So people couldn't really use this Scrappy algorithm and really required manual tweaking so that you can really achieve final accuracy, better accuracy. And with error correction, what we realize is that you can use all of these standard segmentation algorithms and apply error correction and still achieve a much better accuracy. So basically, this eliminates the requirement for really manual tweaking of these prior algorithms so that they can work with the newer generation. In terms of also the overhead and the runtime that our HMM-based work introduces over the tools, so we see that Crane introduces negligible overhead, especially for larger genomes, right? So the overhead is a bit higher for smaller genomes such as E. coli. So this means actually the bottleneck really shifts with increasing genome sizes. So this work was recently accepted for a talk at ISMB in the HeatSeek track. So if you're attending ISMB, please stop by and hear Simon's talk on this direction. And before I conclude, I just want to show one more thing. This is a work in progress, but also there will be a poster about this at ISMB. So if you're curious, you can learn more about it. But I want to show is that, so remember earlier, I showed you that the DNA or our genome is a series, so it's a basically huge string of DNA letters. But I sort of lied to you. It's basically not just DNA letters. It also contains certain, let's say, modifications that we call base modifications that can happen at certain positions, which will change the structure of the DNA. And this is extremely critical because it's known as it plays a critical role, important role, especially, for example, for cancer. And of course, what people do, people develop tools, and there are basically different modification types, different modification profiles. And people design tools, these are mainly ML tools, to identify each modification type, specifically basically trained for a particular type, right? And what we wanted to do is that we want to design a protocol that can actually identify all different base modifications in maybe like sort of in a label-free way, so that you don't really have to train your model for each different modification type every time that you want to analyze them. So for this, what we did, some basic signal pileup so that we can understand the structure of the signals when there's a base modification. So this is a very simple example of it. So here, for example, you see like the signals seem to be agreeing with each other, but at this point, there is something weird going on. And then we basically generate some features out of the things that we're seeing in the signal, and designed, integrate all these features in a simple UNet architecture so that we can label each position either as modified or unmodified. So our initial results provide a significantly high accuracy, especially compared to the prior approaches. But I don't want to give you any further spoilers. So if you're interested, you can, if you're attending ISMB, you can talk to Bargao about this. So to conclude, I think there are really still interesting challenges in signal analysis. So we should be better integrating the chemical structure information in genomics, right? So how can we identify the novel base modifications by directly analyzing signals? Or maybe we can still use this noise information to do some useful stuff. Also, we need to be redesigning effective solutions for storage and also adaptive computing as the requirement of the computation changes. And also, we need to be enabling end-to-end energy-efficient analysis, especially for portable sequencing devices. And today, I only covered maybe scratch the surface of our research that we're doing in our group. So we're also interested in co-designing the architectures and also AI solutions and algorithms for analyzing genomic data, especially for large volumes of genomic data. So with this, I conclude and happy to take questions. Thanks.
Thank you very much, Jan.
Hi, thank you for the talk. I'm not super expert in the machine learning or the number field, but do you think that trying to be adaptable in terms of, for the quantization part, sorry, you say that having a fixed number of buckets of beans basically is a kind of limitation that requires you then to add your further step of the pipeline. Do you believe that adding something that is adaptable in terms of binning capability could, I don't know, remove the hash or have introduce some benefit? I don't know, just out of curiosity.
So here the adaptability is then about the other parameters in quantization, right? So the number of bins that we can have perhaps, and the bits of those bins.
Yeah, basically while you're doing the acquisition, you have a method that adaptable in an adaptable way detect if your distribution of data changes, and then changing the distribution, you need a different number of bucket to kind of have less or I don't know noise maybe, I don't know.
Right.
I'm not an expert, just that I know a method for that, and I was curious if it's something worthy or not.
Right. Yeah, I think being adaptable is definitely very important and is definitely worth for the analysis. I think we need to analyze to understand the requirements of the analysis. For example, if what you want to understand whether a read is coming from a certain genome, for example, right? So what these quantizations or this adaptive quantization is designed for is that you identify the mapping position at the base level in the reference genome, right? So that's why they are still at the final granularity. So they cover, for example, about 10 to 13 bases in the read, right? But then the next question is, do you want to know maybe the mapping position like at the base level, or do you want to know if the read is coming from a certain genome or not? The second question is the granularity of that second question is larger, right? So in that case, perhaps what you can do, you can maybe pack more, right? Pack more quantized values, or maybe either increase or decrease the number of bits that you're assigning for each quantization and so on, right? And by doing so, the next question becomes, so how many you can pack? Do you have a like 64-bit variable, 128, and so on, right? So all of these questions are really about adaptability, and it really impacts the performance and the accuracy of the analysis that you're doing. So in short, yes, I think it's worked.
Thank you very much.
And anything else? Okay.
For CERN or Crane, have you considered using an end-to-end neural network? Would that be possible, or you have to rely on hidden Markov models to some extent?
This is actually a very insightful and good question. So we actually did, in terms of accuracy, accuracy can also potentially is better in certain cases when you're using neural nets compared to HMM. But the issue was the computational efficiency. So the reason for that is the segmentation itself in the raw signal analysis is only 2% or 3%. And if you increase that runtime to something like really, really large, it starts taking up almost the entire execution time, which creates a huge problem for these type of tools. So the main limitation for us is really, since the original segmentation is really cheap, we don't really want to go. We don't really want to be much slower than the cost of original segmentation. If you can come up with maybe either with an architecture or hardware that can do this neural net type of analysis, I believe that it will even lead to much better error correction in terms of accuracy, while still being faster. But while being right now, as we're being in the CPU paradigm, we need to be thinking about this runtime. But going forward, right, we need to think about those architectures, and in terms of hardware and also ML architecture. Yeah.
This actually, maybe shortly, I'm going to ask something too. What about plans about accelerating then, Rojas, in hardware?
Maybe your talk is going to talk about this, right? So it's a really nice transition maybe to your talk. But yeah, I think there are really good opportunities, especially knowing that this is a portable device. So we should.
It's my intention,
Yeah.
because you started your talk today talking about devices that can be used at home. So basically, it would be interesting to see if this particular algorithm could be actually embedded and portable and used.
So one important metric is the energy that maybe most people overlook.
Thank you very much, Jan.
Thanks.
Okay, so last one. I'm gonna try and keep it short. Yeah, thank you. I mean, you add a microphone. I think Jan is ready.
Quickly, a little bit about Konstantina as we wrap up the workshop. Konstantina is a postdoc researcher at Safari, led by Professor Onur. She has a PhD from National Technical University of Athens. Her research interests are in the field of computer systems and architecture, heterogeneous computing, and hardware acceleration.
Thank you very much for your introduction. Was it sharing already? Is it online then? Okay, I got it. It's not. Cool. Okay. So thank you, everyone. Good. Okay, so everyone, I'm going to try to keep it a little bit short. I'm going to change a little bit the perspective and talk again about acceleration, but this time we're going to include the aspect of the I/O overhead. I'm going to try to skip through the background because it has been covered mostly by Jan already and Onur and the previous talks. So we've seen the importance of genome analysis and that it can lead in very important breakthroughs and hopefully more and more into personalized medicine. So we've seen so far that genome analysis typically starts with two major steps, which is the sequencing part and the mapping part. And we saw that one of the approaches that has been most the traditional approach is to use base calling, which is based on deep neural networks, to translate the signals into bases and then continue with a downstream analysis. Although this brings us very accurate results, it has the downside of being particularly costly and power hungry. So this has led to the raw signal analysis that maps signals directly to the reference genome without doing the base calling. So this can lead us to efficient analysis that can scale better and even be portable, and potentially enable new analysis that, since the raw signals include some more rich information. So this talk is going to focus on the raw signal genome analysis. It is a novel approach that cooperates directly on the raw electrical signals. It can explore the high throughput of the modern sequencing technologies and enable real-time analysis. So we saw a little bit of the workflow before with Jan in details. There is an offline part that is the indexing part. So we first need to translate the reference genome from the basis to a signal, and then use some sketching and hashing mechanism to store small parts and segments of the reference into a hash table that will allow us to do some very quick lookups of the signal. Then we have the mapping. The mapping is happening online, and it's basically used to use a sequence and find potential matching locations in the reference genome. So in raw signal genome analysis, we can focus on three major steps, which is the event detection, the seeding, and the chaining part. During the event detection, we use again some sketching mechanism to split the query into shorter segments or fragments that were called events. And then we're using a compact set of events to query after hashing the event, we're using it to query the hash table and find potential matching locations in the reference genome. Then in the chaining part, we're using these matching locations to form some consistent and collinear sets of them that can describe estimate our alignment. So this last operation is focused mainly on sorting and dynamic programming. So this is very much computational intensive. So let's go to the motivation and the goal of this work. So we've seen that sequencing throughput is rapidly increasing, and it's going to keep in that trend in the future. And raw signal genome analysis struggles to keep up with the increasing real-time requirements. Therefore, there is indeed a need to accelerate the workflow. So what we need to do is to identify the key performance bottlenecks to do this efficiently. We follow this approach, a typical approach, where we have to identify which is the most time and energy intensive parts of the pipeline. So we're going to test 5 different data sets, starting from smaller genomes, from D1 data set here to D5, which is a much larger genome. And we're going to see the time breakdown of the application. We're going to check how much time event detection, seeding, and chaining takes. But also, we're going to look into the I/O overhead. So we can see here in the time breakdown that, as expected, the chaining and seeding part consistently dominate the runtime, but also the event detection and the I/O overhead are not negligible. So in a conventional approach, we would indeed target the chaining and seeding to accelerate. But what will happen to the rest? What will happen after that, basically? We're going to model here the I/O overhead to see what happens to it when we continue to when we accelerate the seeding and chaining, and what happens when we increase basically the speed up efforts, so minimize the latency of the seeding and chaining. So eventually we can see that I/O becomes the major bottleneck since the other computational intensive parts have been diminished. So therefore, we have the goal to able to design a high performance and energy efficient system for raw signal genome analysis. We have to address both the I/O data movement bottleneck as well as the computational bottlenecks. So this is what Mars tries to achieve. So what is Mars? Mars is the first in storage processing system for raw signal genome analysis. The key idea is to create an in-storage processing system for raw signal analysis that can address the I/O data movement bottleneck, but also exploit the parallelization capabilities of SSDs to accelerate the computational intensive parts. So it is a co-design effort that combines targeted software modification as well as some dedicated hardware components within the storage system. The software modifications aim to optimize raw signal genome analysis for use inside the SSD and also leverage the parallelization capabilities within the SSDs, while the dedicated hardware components are actually components that are developed using processing in memory paradigms. For example, processing using DRAM and processing near DRAM by using the internal DRAM of the SSD. So let's see a little bit first of the software modifications that we explore. The software modifications have 2 objectives. The first one is to decrease indeed the computational workload, and the second one is to also decrease the intermediate storage requirements. So first, we're going to explore some filtering techniques. The goal of them is to discard candidates that are not going to actually lead into useful alignments. And we leverage frequency-based filtering as well as vote-based filtering. The second part is taking advantage of the hardware capabilities and using arithmetic conversions of fixed point instead of floating point, which will both reduce the memory footprint as well as the computational time that takes for our operations to happen. Now, what we need to make sure before we proceed with architecture is that we can, after applying these modifications, we preserve the accuracy despite the inherent raw signal noise. So we managed to achieve that by carefully placing these software modifications and extra steps into the pipeline, and also carefully fine-tuning the parameters of the filters. So we're going to validate this. So we're going to compare with a ground truth that is a typical conventional based pipeline that does the base calling with Dorado and the mapping with Minimap2. We're going to use the same data sets, and we're going to compare with the state-of-the-art software for raw signal genome analysis, which is ROHAS. and two variants of our design. The first variant only includes the filtering, and the second one includes the filtering plus the arithmetic conversion. We can indeed validate that by adding our filtering techniques, we effectively eliminate some ambiguous and redundant matches and manage to focus on more informative regions. So we managed to even improve the recall and F1 score in comparison to Rojas 2. And second, we can observe here that when we leverage the fixed point arithmetic, we don't have any significant loss. So it's worth a trade-off. So now let's move on to our architecture. So we have the first storage processing system for raw single genome analysis. The SSD now can operate in two modes. In one mode, we have a conventional use, where the SSD performs all the standard SSD storage operations. And in accelerator mode, SSD can actually run the raw single genome analysis pipeline without needing any intervention from the host. Now we're going to see a little bit one by one the components that we create. First of all, we're going to focus on the SSD controller components. We have the Mars control unit. This is basically a finite state machine that orchestrates the pipeline execution, the control, and the data flow. We also create on the flash controller two units, the sorter unit and the merger unit. The sorter unit is specialized to perform parallel sorting of sequences by using some dedicated logic. It can support up to a fixed length of sequences, and for that purpose, we accompany it with a merger unit that can combine efficiently some sorted subsequences into longer sorted ones. These units operate on data that are coming from the internal DRAM, so it's basically uses processing near DRAM paradigm. Now let's go on to the components that we instantiate inside the internal DRAM of the SSD. First, we have the arithmetic unit. The purpose of the arithmetic unit is to perform arithmetic and logic operations from all steps required for FPGA. So this is basically the processing near DRAM paradigm, and it's based on an existing fulcrum architecture that leverages the proximity to the DRAM subarrays to perform these operations with low latency. Next, we have the querying unit. This is done with processing using DRAM, and the purpose is to perform the lookup operations very efficiently. For that reason, we leverage another existing work that is called Pluto, and it leverages the DRAM row activation to do massively parallel lookups on the hash table. So what we do is that we transform the raw signal genome analysis steps into some more fine-grained tasks that are actually in line with the operations that we have defined in our components. So we have to describe only arithmetic, querying, and sorting operations for the whole pipeline. And then we express our Mars pipeline in the terms of these tasks. Now, each of these tasks can be mapped in one of the dedicated compute units that we have. For example, if we have a multiplication in any step of the workflow, this is now going to happen on the arithmetic unit. So therefore, we have the control unit. This is a finite state machine that encodes this pipeline into a finite state machine. It's a fixed execution sequence, but when we have our data incoming, they are dynamically triggered based on the input. So some of the steps, for example, could be, let's say, bypassed. So let's go quickly now through the flow of data and the control through this entire architecture. So initially, our data is stored in the flash memory. We're going to fetch them and move them to the internal DRAM. So we have our own input signals, and we start with the event detection that happens in the arithmetic unit. Next, we need to go to the seeding part. We're going to use, therefore, first we're going to create our hash values and do the filtering. This happens again in the arithmetic unit. Now, once we have these smaller hash values, we're going to query the hash table, and this is going to happen again using the DRAM with the querying unit. Now that we have the matching positions available, we're going to proceed with the chaining part. First, we're going to create some batches of sequences, but we're going to move them to the SSD controller in order to do the sorting. If it is required, we're going to also do some merging of the longer sequences, and we're going to return back to the internal DRAM, the sorted to finish the chaining by using dynamic programming. Now, there are much more details on how this is done and how we preserve also the typical SSD operations, that you can check them out more in the paper. Now let's go to the evaluation. We're going to compare our system with 4 other systems for the performance and energy analysis. First, we're going to use a typical conventional pipeline that performs the base calling by leveraging GPUs. So we use the Dorado framework and the read mapping with Minimap2. We're also comparing with hardware-accelerated typical base calling-based pipeline, which is GenPip. And for the software we're using, for the raw signal analysis domain, we're comparing ourselves with RoHS 2, which is the state-of-the-art CPU-based pipeline. And then we have the software-only version of our tool that only incorporates the software modifications. And last, we have our own system, Mars. Let's see some parts of the experimental setup. We're using a state-of-the-art server that has 2 AMD CPUs with 64 physical cores, 1 terabyte of DDR4 DRAM. And for the part that can actually be offloaded to the GPUs, which is basically Dorado, we use the NVIDIA RTX A6000 GPU. For the SSD, we have 8 channels with each one of them 8 flash chips and PCIe 4 interface. And the internal DRAM has 4 GB of DDR4 with 16 banks and 5 12 subarrays. All the dedicated hardware that we create, we use Synopsys to create it. And for emulating the storage components, we use MQSIM, while for DRAM, Cacti 7. So let's see here our results. The performance evaluation, we can see that Mars indeed outperforms all other baselines across all data sets. And it's some insights on the results is that Mars can indeed outperform GenPip, which is the hardware accelerated part that adopts actually processing in memory. But this happens outside the storage. And this highlights the fact that Mars can indeed fundamentally solve the I/O data movement overhead. Also, it's very interesting to notice that we already gain a 1.7 speed up over RoHS 2, which means that also the software modifications can allow for some significant speed up. And I think it also highlights the fact that the co-design approaches always leads to better results. Similarly, we have reduced energy consumption over all other baselines. And in this step, I would like to highlight the fact that this energy reduction does not only come from reducing the execution time of the entire pipeline, but also by the fact that we actually avoid some needless data movement overheads. So let's now conclude. Mars was the first in storage processing system for end-to-end raw signal genome analysis that is enabled through both software improvements and leveraging processing in memory inside the storage. We managed to improve performance against software-based, base calling-based pipelines, but also hardware-accelerated base calling-based pipelines, as well as the software-based raw signal genome analysis state-of-the-art. We managed to keep the accuracy and even improve it in some cases with significant performance and energy consumption reduction. So this was Mars. Thank you so much. I'm happy to take questions.
Hi, thank you for your presentation. Out of curiosity, one is a technical question is, I don't know if, do you use a GPU with DPX instructions in this comparison? And I don't know if it's useful in any way, honestly. I don't think so, but...
I'm not really sure about that.
Okay. Yeah, no problem. It was just out of curiosity. And the other thing is, the results are impressive in terms of performance and energy consumption. So do you believe that with Mars, one could envision that you build a portable SSD raw signal? I don't remember. I just remember the acronym. Thank you. Sorry.
Well, this was part, I think, of the. It is part of the idea, and we would like to go towards that way of embedding it. I think there is still a long way to go because there is also to take into account how you store the data from the beginning into storage. So you have to take account in some other basically overheads to make sure that from end to end, you're going to have some significant speedups. But I think it's very promising. And yes, as I asked some before, I think we should work to actually have it in a portable device. You're welcome.
All right, any other questions? If not, I think
We
we've
can conclude.
come to the end of the workshop. Thanks to all the
Some-
speakers. Was a good interaction. Looking forward to the next event. Thank you.
Thank you, everyone. Onur, do you want to say something for the finish? For good? It's true.
This tea helped, but it was discovered too late. Okay,
Yeah.
both.
No, mine.
Okay. No, take the one that you don't like then. Okay, it's good to have everyone here, although I had to step out for the other workshop. I hope we can have another one with more engagement from industries who can actually integrate some of these solutions to their systems, including medicine and other places in the future. So thanks for coming. Thanks for staying until the end also. And don't get sick.
It's extremely cold.